Greetings all. Thank you very much for coming back for today's session. We're going to start sharp. We're as sharp as we can at 9 o'clock to try to keep on track. And hopefully all of you will hang in for all of the excellent sessions and also for the breakout sessions and the roundtable this afternoon, as we really value all of your inputs. I'm going to turn it immediately over to Dr. Jeff Hall, who will be our chair for our first session. Take it away, Jeff.
Thank you, Stephen. For those who don't know me, I'm the Associate Director of Education and Outreach at the BD Water Research Center. Our talks this morning, the first session, will be on water security, equity, and supply, obviously a huge concern around the world. Just a few reminders for everyone. Please keep your microphones muted if you're not speaking. Each speaker will have 15 minutes. I'll jump on if it looks like you're going to be going very close to that for a quick reminder. If you have any questions, put them in the chat. If we have time at the end of the talk, we'll try to answer those. Otherwise, the speaker can come back to you by chat and answer those. So our first speaker is Tessa Latchmore from the School of Environmental Studies. Her title is Using a Large Spatiotemporal Groundwater Quality Dataset to Delineate a Suite of Quantitative Microbial Risk Assessments for Private Drinking Water Wells in Ontario. So take it away, Tessa.
Thanks, Jeff. I'll just share my screen here. Can everyone hear me? Yep, okay. Maybe. Sorry. Ooh. Which screen are you seeing?
That looks good.
Is it just the slideshow?
Yes, that's perfect.
Okay. So hi and good morning, everyone. My name is Tessa Latchmore, and I am this is just my way here. I am a PhD candidate in the School of Environmental Studies at Queen's University. I'm supervised by Dr. Anna Majuri, Dr. Paul Hines, and Dr. Stephen Brown. Today, my presentation is going to focus on using a large spatiotemporal groundwater quality dataset to delineate a suite of quantitative microbial risk assessments, or QMRA, for private drinking water wells in Ontario. So just a bit of an outline. I'll be discussing background, so just private well water in Ontario, as well as how I will be using detection rates. I'll look at the methods, some of my findings and conclusions and next steps. So rural households in Ontario depend almost entirely on groundwater from private wells, with approximately 500,000 domestic wells serving 1.5 million individuals. Unlike municipal drinking water systems, private drinking water wells are not required to meet the regulatory standards under the Ontario Safe Drinking Water Act or the Ontario Clean Water Act. Therefore, the well owner is responsible for the condition and maintenance of their private drinking water source. Studies have shown that contamination events may be particularly pronounced in rural areas where increasingly intensive agricultural activities may combine with urban growth to threaten the quality of private drinking water supplies. Previous research in Canada has shown that populations dependent on private water supplies are characterized by an increased risk of gastrointestinal illness versus those using municipal water systems. However, there's a difficulty in quantifying the health burden associated with private drinking water systems. Often individuals who experience adverse human health effects such as acute gastrointestinal illness do not seek medical care as it is mild or self-limiting. Additionally, contamination events are often chronic or sporadic, with cases largely underreported or misdiagnosed. Quantifying waterborne illness is further complicated given that some residents may acquire immunity as a result of ongoing or historical exposure to contaminants, but this of course has implications for those visiting or who have never been exposed to the water source before. So one way to quantify the effect that private water wells have on human health is through risk analysis. The primary focus of my current project, and I guess this presentation, is risk assessment, specifically a quantitative microbial risk assessment, or QMRA. But I did want to put it in the context of risk analysis, which consists of risk assessment, risk management, and risk communication. I really won't go into too much detail on the other processes, but it's important to note the double-sided arrows highlight the continuous cycle of risk analysis, so any changes or improvements in one area are reflective in another. So in order to create a risk assessment that accurately reflects private well water in Ontario, I examined detection rates because an understanding of E. coli detection in private well water and the subsequent effects of sampling frequency can assess with the development and space sampling strategies for well owners, health risk assessments and interventions to ensure adequate human health protection from waterborne pathogens. So ultimately, the goal in using detection rates is to identify potential contamination pathways to inform QMRA delineation if needed be. So detection rate is defined as the proportion of E. coli positive wells. So the number of wells in which E. coli bacteria are detected by the number of wells in that sample population. This was previously defined in 2015 by Atherholt, who looked at detection rates in private well water in New Jersey. But detection rates vary significantly due to natural fluctuations of indicator bacteria in groundwater. Also because indicator bacteria like E. coli are usually detected sporadically and in low concentrations in groundwater. Another factor that can influence the detection rate is the number of times well is sampled, and which is the focus of this presentation. Just taking a little bit of a step back, the overall aim of my PhD work is to provide a novel and holistic assessment of the human health burden associated with unregulated private drinking water in Ontario by examining the social and physical factors that influence community water security. But more specifically today, I'm going to be speaking more so about delineating those risk assessments so they are increasingly accurate and spatially specific, because in reality, there is no one size fits all risk assessment that can address all public health issues, problems, and regulatory needs. So well water submission data from Public Health Ontario from 2010 to 2017 was analyzed for E. coli to study the relationship between sampling frequency and E. coli detection in Ontario private wells. Detection rates were further analyzed relative to geological setting, season, and subsoil and well depth. Bivariate statistical analysis was performed when comparing greater than 2 groups of detection rates and to quantify the association between groups. Each well was grouped based on the number of times it was sampled, the number of times it was tested on an annual basis, so 1 to 5 times a year, with each well being assigned an individual detection rate. Wells sampled on 6 or more occasions were actually excluded from the analysis. While high sample numbers are useful, they're not necessarily representative of the testing frequency across the province, and they may introduce a bias due to excessively high or low detection rates depending on sampling motivation. So I use well water submission data from the well water testing data set, which is maintained by Public Health Ontario and included approximately 700,000 samples from 240,000 wells from 2010 to 2017. Also, just a note, it's completely voluntary to test your well water in Ontario. It's also free at the 11 provincial laboratories, but although it is not required, it's definitely recommended. So this illustrates the proportion of E. coli positive samples in each year in the data set separated by region in Ontario. So Eastern, Western, Central, and Northern. And this is the number of submissions, not unique wells. As you can see, 2016 is noticeably lower, and this was actually a drought year in rural Ontario, and it's highest in 2013, and this year was characterized by a lot of localized flooding. The average rate of bacterial contamination in these submissions range from 2.3% to 4.5%. And to compare this, a study also done with Dr. Anna Majuri at Public Health Ontario found a bacterial contamination rate of 3% to around 4.3% from 2003 to 2012. So relatively the same. Using data from the Well Water Information System, which is maintained by the Ministry of the Environment, Conservation and Parks, as well as Ontario Geological Survey data, I separated the wells into consolidated and unconsolidated aquifers. Consolidated was denoted as having a bedrock present, so limestone, sandstone, shale, or granite, and unconsolidated aquifers had no bedrock present in the formation material and consisted of clay or other loose materials such as gravel. Over half of the wells present in the data set were located in consolidated aquifers. Detection rates were then calculated based on aquifer type and then compared. It was found that detection rates in consolidated aquifers were significantly higher than unconsolidated across the 7-year period. Once the wells were separated into consolidated and unconsolidated aquifers, I further delineated the data based on depth to bedrock or overburden for consolidated and also well depth for both aquifers. The 4 delineations presented here, so Q1 to Q4, are based on the interquartile range of the data. Shallow and moderate overlying subsoils and well depths were associated with significantly higher detection rates in wells in Q3 and Q4 delineations. In fact, consolidated aquifers exhibited consistently higher detection rates compared to unconsolidated aquifers over the 8-year study period across depth delineations. So within consolidated aquifers, over 50% of the wells were geographically associated with limestone, with the rest associated with granites, sandstones, and shales. Additional geological formation was present in the data set, but it was excluded due to small sample sizes. Annual E. coli detection rates were highest for wells located in limestone, and bedrock specific detection rates were highest during 2013, which was the recognized flood year. So this ranged from 1.5% to 20% for sandstone, for example, for well sampled 1 to 5 times. In limestone, the same sampling frequency during 2013 resulted in detection rates of 3% to around 24%. So E. coli detection rates differed significantly with respect to sampling season. Rates were markedly higher during the summer and fall months compared to spring and winter. More specifically, private well sampled 1 to 5 times during the summer months had E. coli detection rates of 3% to around 36% respectively. However, in the winter months, that ranged from 2% to around 15%, so much lower. Finally, to assess the effects of repeat sampling, I applied sort of basic probability estimates using power analysis to spatial and temporal E. coli detection rates. A summary of these estimates within each respective delineation is presented in the decision tree that you're seeing there. For wells sampled 3 times, which is denoted with a 1, 12 times per year, which is denoted with a 2, and 24 times per year, which is denoted with a 3. So just to walk through this decision tree a bit, sampling during this, sampling during the fall and summer months in shallow and moderate depth wells, probability estimates indicate an E. coli detection of 10%. Of 10% if all wells were sampled 3 times per year. This would increase to 50% if those wells were sampled 24 times per year. However, if sampling occurred during the spring and winter months, E. coli would be detected in 7% of wells and 38% of wells if that increased to 24 times per year. Just to compare this, for shallow and moderate depth wells in unconsolidated aquifers, E. coli would be detected in 12% of wells if each well was sampled 3 times per year, increasing to 56% of wells if each well was sampled 24 times per year. And during the winter months, the same sampling regime would detect E. coli in 4% of wells and 22% of wells, respectively. So ultimately, this provides further evidence of the level of variation associated with regionally specific factors such as season, depth, and geological formation as they pertain to E. coli detection. Additionally, this type of analysis or decision tree, so to speak, may also be used to inform guidelines for well owners regarding the most appropriate time to sample their water supply. So for example, if an individual was to sample their well 1 to 3 times per year, which follows the guidelines from many Canadian provinces, sampling during the summer months may provide a more realistic picture of their overall well water susceptibility than sampling during the winter and spring. So overall, well water contamination is complex and it's dependent on a multitude of factors from the well owner's behaviors and practices to the physical characteristics of the wells and the environment in which it is situated. So it's crucial that we have recommendations and guidelines that are reflective of these source-specific factors. The spatially specific findings that I outlined illustrate the influence of hydrogeological setting on E. coli detection in Ontario private wells. However, to date, few, if any, human health risk assessments of waterborne infection have accounted for these spatial delineations. So more specifically, moving forward in my PhD work, I'm going to be creating a risk assessment for private well owners located on consolidated aquifers and one for private well owners located on unconsolidated aquifers. So ultimately, delineated health risk assessments based on spatial drivers may provide an increasingly accurate estimation of human health burden and may also assist in the development of evidence-based guidance for well water sampling and testing. Thank you.
Okay, thank you, Tessa. That was a fantastic presentation and a great way to start our morning. And your timing is perfect.
Okay.
We don't have any time for any questions, but if anybody does have follow-up questions for Tessa, please feel free to contact her. Our next speaker is Lauren Halliwell from the Department of Civil Engineering, and the title of her talk is Wastewater Issues, Treatment and Effluent Release for Small Municipalities. So Lauren, take it away.
Thank you, Dr. Hall. Let me just share my screen. Can you see my presentation?
I see the non-presentation version.
Yes. Okay. So there we go.
And you might want to flip your presenter mode.
Sorry. Okay.
There we go.
Good. Okay. So hi, everyone. My name is Lauren Halliwell, and I am currently upgrading to a PhD, but I'm currently in my Master's of Applied Science here at Queen's University in civil engineering, specializing in environmental engineering. My supervisors are Dr. Pascal Champagne, Dr. Jeffrey Hall, Dr. Hamadriza Shirkenny, and my collaborations on my project are with Loyalist Township and the National Research Council of Canada. I'm here to talk to you today about my topic, which is wastewater issues, treatment and effluent release in smaller scale communities and municipalities. So as you can see on my screen here, the Canadian Environmental Protection Act of 1999 states that an act respecting pollution, prevention, and protection of environment and human health in order to contribute to sustainable development. And so basically what my point is here is my talk is basically focusing on wastewater treatment systems in Canada. As you can see here, there's this river is contaminated with fecal bacteria. This is just one specific example that I wanted to bring to your attention, which was in 2015, the Le Havre River in Nova Scotia, which focused basically in the news on a water treatment system that released gallons and gallons of wastewater treatment or wastewater into the Le Havre River. So that just is one example. This has happened all over Canada and happens often because wastewater treatment systems have to be updated. And so basically that's kind of a call to action as to how engineers and environmentalists need to bring to the attention of basically benefiting these systems and updating them. And so this is where I go into my, sorry, this is not working. Okay, there we go, sorry. And so that brings that into my research topic, which is on my master's, which is looking at wastewater stabilization ponds. So there's different types of wastewater treatment systems in Canada. There's primary, secondary, and tertiary treatment. These are basically in succession, but there's different types of secondary wastewater treatment systems. There's chemical, but then there's also sustainable. And so in Canada, there are things called wastewater stabilization ponds, which are prevalent in specifically smaller scale communities because they're economically feasible and they're also operationally feasible. So they're used for smaller scale communities because they use natural wastewater breakdown processes in the environment to break down municipal wastewater. And so as you can see here, which is my case study for my master's research, which is Amherst, Ontario, this is a traditional wastewater treatment system where it starts with anaerobic ponds, which is the first pond. Then it goes into facultative, which utilizes biological oxygen demand to break down the wastewater. And then the water flows in succession to a maturation pond, which uses UV disinfection and in a shallower pond to break down the wastewater further. in the Amherst wastewater treatment system, which is just outside of Kingston, about 20 minutes away. You can see here a picture of an aerial view where the operator building takes in from the primary wastewater treatment. Then it sifts the solids and the liquids into different, the solids basically leave into a different plant, and the liquids continue through secondary wastewater treatment. And so this is the first pond. You can see the facultative pond, the maturation pond, and thirdly, which is the newest addition to this wastewater treatment system, which is the constructive wetland, which is a basically series of cells with natural native macrophyte species from the area that further break down the wastewater treatment system. So on the left, you can see a traditional diagram, which is given in wastewater treatment engineering courses in undergrad, even here at Queen's, which explain how to design a WSP, which is wastewater treatment pond. But on the Amherst side of things, they were finding that there was high pH levels. So they additionally added the constructive wetland to mitigate those levels. So engineers are continuously trying to design these wastewater treatment systems so that they're not, so they're meeting regulations in Environmental Protection Act, because this water basically is released into the natural ecosystem around the area. For example, after the pond 3 there, you can see it's all green area. It's actually a golf course, and then later, like I think maybe a 1-minute drive down the road, there's a natural lagoon that's just down the road. So that could be a huge issue if the water is released and not meeting those regulations. It could be a human health issue because wastewater can have harmful bacteria like E. coli, but it also could be harmful to the natural environment in the surrounding area for terrestrial and aquatic ecosystems. And so my project specifically is basically honing in on the gap of specifying what these water quality parameters are, how this wastewater treatment system is breaking down these water quality parameters so that we can really understand what's happening and meet those environmental regulations. So this pond specifically, Amherst does meet these regulations, but there is yet to be a numerical modeling tool to compute out how to design this system really specifically, that couples the water quality components, which is all of the substances. So you can see on the right here, dissolved oxygen, carbon oxygen demand, ammonium nitrate, etc. And also coupling it with the hydrodynamics, which is all incorporated in the process of this system. And so that would be the ambient temperature, as well as the water velocities moving through the system. So my goal of my research project is to couple this hydrodynamic and water quality model so that us as engineers can basically look at the system and look at the nitty-gritty details of how it's working throughout the system, so that, for example, as this case study for Amherst, we can use this to better design these treatment systems, but specifically better design them in the face of climate change. So basically my role is to manipulate the Environment Climate Canada patterns and see how the water system will basically treat this wastewater. My third goal of my project is also to create an ice cover winter model. So in the face of climate change, winter is the most susceptible season to varying effects due to treatment systems essentially over the winter time in Canada are assumed to just be frozen over and essentially not to work. So you would just only account for the time period of treatment over the summer times and the warmer seasons. And so there has yet to be a numerical model that would help to understand the processes in terms of partially freezing winter. So here in Southern Ontario, the ponds will be partially frozen /frozen and back and forth throughout the winter season. And so, to just give you an understanding of how this numerical modeling tool will work, is it basically computes a calculation for each one of the model substances and calculates it for a small grid cell, and does that for the entire process of the pond, which gives you a really good idea of how each step of the water moving in succession will be treated throughout the wastewater treatment system. And so my role as a scientist here was to go into the field and collect actual data to compare to my theoretical data for my numerical model that I'm creating. And so I did this over 2 seasons, summer 2019 and winter 2020. I'm also running another season this winter as well, and hopefully again this summer. And essentially I took water samples throughout, I took water samples throughout the whole summer season, collecting all of this data on the left and all this data on the right. I'm not going to run through every single detail, but the reason why I added it there is so that I was really trying to get a thorough understanding of what's happening in the pond, and also so that I can really understand comparing my theoretical results of what I want to compute out of the model. So basically, I'm putting into the model these values such as like temperature and velocity values, but I wanted to see the outputs of the model and compare it to what was actually happening in the pond to validate this model. And so I took various samples, and then also over the winter time, I also took a bunch of samples as well. And so this included dropping water temperature sensors in the winter time, boating over top of the wastewater treatment pond and dropping these sensors to get these calculations, so that ultimately I can compare my results to what my model output will be. And so my current state is, been quite a long process, but it's been very much looking at how the model is working, what the calculations look like, and how my boundary conditions, so the upper, which is the values going into the pond, are comparing to the actual values, and then what the computations coming out of the model look like. And then also looking at my ice cover model. So I'm currently in the process of looking at that winter data analysis and completing my winter model calculations. So that's using a numerical code to input into the model to compute out using spatial, so the amount of ice that's covering the pond, and calculating where that ice would be covered, showing aerobic value or aerobic conditions underneath the ice. And then around the area that it's not frozen, the anaerobic conditions of the processes that are naturally occurring. And so just to bring you to an understanding of why this research is necessary, especially for water treatment and water quality, it's ultimately important to have better engineering designs as climate change is impacting all systems, all water systems, especially wastewater treatment. And this not only incorporates social, so communities surrounding the area of human health, it incorporates environmental and economic for smaller scale communities, especially these water treatment systems that are used in, for example, Toronto compared to a municipality of like 1,000 or 2,000 is quite different, and the budgets and economic stances for this is very much in need of updating these wastewater treatment systems. But we need a better operation of what kind of treatment systems are implemented, because they need to be economically feasible for these smaller scale communities. And so having a numerical modeling tool like this specifically for Amherst-View can be a baseline case and used for implementing these water treatment systems into other smaller scale communities that are in need of updating their wastewater treatment systems. For example, Indigenous communities that often are kind of slide under the radar of having improper wastewater treatment systems and leaching wastewater treatment into their drinking water. And so this type of pond can be used hopefully as a sustainable wastewater treatment system for these communities, but also understanding the implications of these wastewater stabilization pond designs in the face of climate change, so that we can better safeguard human health and the environment. Thank you.
Well, thank you, Lauren. That was another great presentation, and our timing is pretty much right on. So if anyone has any questions for Lauren, please feel free to put them in the chat, and she can be watching that to answer. So we're going to move right along. Our next speaker is Sophie Fulford from the Department of Civil Engineering at Queen's, and her talk is titled Early Detection of SARS-CoV-2 in Municipal Wastewater. So Sophie, please take it away.
Can everyone see that and can everyone hear me?
Yes.
Okay, awesome. I'll get started. All right, so thank you, Jeff, for that introduction. As you mentioned, I'm a PhD candidate in the Department of Civil Engineering at Queen's. Today, I'm going to be talking about early detection tools for SARS-CoV-2, which is the virus responsible for COVID-19 within municipal wastewater. So for today's presentation, I'm going to be talking a little bit about the context of the pandemic that we're currently in from the start in 2019 to present. And I'll be talking a little bit how people are using wastewater-based epidemiology to be able to detect the virus in wastewater as an early warning system. And I'm going to be talking about a couple of studies worldwide and also what's being done in Canada. And then I'm going to transition to the work at the BD Water Research Center that we're currently getting started and optimized for future use. So I think nobody's going to be too surprised by these statistics, but since 2019, the coronavirus has spread to 219 countries or territories. It's caused over 106 million infections, over 2.3 million deaths, and in Canada alone, it's at almost 1 million infections and over 20,000 deaths. So a lot of us will think of the respiratory route as being the primary pathway of concern, but it's not the only potential transmission pathway. So SARS-CoV-2 has also been detected within the stool of infected individuals. This is not just people who are symptomatic for the illness, but all of those, also those before they get symptoms, and then those who don't show any symptoms at all. So the thought is, and what a lot of people have been pursuing all over the world, is that the detection of the virus within wastewater can act as an early warning system against future outbreaks for high-risk communities, and potentially for detecting other variants that we're starting to see a lot of now. And so this figure just on the right here is a map from the Johns Hopkins Coronavirus Resources Center. And so as you can see, I mean, it's a pretty widespread virus. It's all over the world, but there's especially dense patches in the United States, in Europe, and as well in this area around India too. So wastewater-based epidemiology is a method that uses chemical analysis of pollutants and biomarkers for the qualitative and quantitative data on the activity of people in a sewer shed. So that's pretty good, pretty well demonstrated by this figure here on the right. So essentially, if you're testing for coronavirus in the clinical setting, you can really only test one person at a time. But if you're going to be using this wastewater-based epidemiology, you're getting sewage from the entire community that you can test all at once. So you're getting it from people's houses, you're getting it from community-wide facilities like shopping malls or restaurants, and you're going to get it from institutions like hospitals, universities, and prisons, and it's all going into the same place. The wastewater-based epidemiology has been used in the past fairly well. It's been used to detect pharmaceuticals and chemicals, as well as other pathogens. So a good actual example of this is that the World Health Organization recommends to use environmental surveillance like wastewater-based epidemiology to detect poliovirus. And in 2013, Israel actually was able to detect it in their sewage samples before they had any reemergence of clinical cases. So worldwide, as I mentioned, a lot of people are already undertaking these efforts. This includes both the US and Canada, as well as countries in South America, a lot of countries in Europe and Asia, as well as Australia. And this is by no means a complete list. This is just a few I've picked out. This is actually a really interesting headline over here to the right. But essentially the University of Arizona, what they did was they tested wastewater in a particular student residence just after a few days when all the students had moved in and they had previously tested negative for the virus. They actually were able to find that a couple people were asymptomatic when they did a retest after they saw it in the sewage. So that acted as a really good early warning, and it potentially prevented an outbreak at the university. So this is just me cherry picking some studies. There are a lot more that have been looking at coronavirus with wastewater-based epidemiology. So I just wanted to get a good geographic representation. And as you can see, a majority of studies did detect the virus in some manner from the results here on the side. Now I'm just going to bring a couple into focus. First of all, I did notice in the literature when I was going through, a lot of studies were able to detect small concentrations of the viral RNA in the sewage, even when there was a low amount of cases in the community. So it was able to pick up on that quick. And then as they started getting to that wave 1 in mid-March and April, they saw that the concentration rise. One example is this article from Edema, and they looked at this in the Netherlands, and that's just down right here. And they found in the beginning of March, only 4 out of 7 wastewater samples were positive for the viral RNA, but in the middle of the March, 9 out of 9 of the samples were positive. This is another early study done by Ahmed et al. In Australia, and that showed very similar results as well. Unfortunately, it's being a bit covered up there. And then, of course, we have work that's going on in Canada. So these 2 studies are from a group out of the University of Ottawa, and they've done some really great work that has shown not only the ability to detect in wastewater, but also that the wastewater was able to identify the viral RNA 48 hours before clinical tests became positive and 96 hours before hospitalizations started to increase as well. And then one other study internationally that I wanted to bring to your attention is this one from Spain. I thought this was interesting because they demonstrated how the samples changed in their viral RNA content as it moved through treatment. So you can see down here, 35 out of 42 influence samples were positive, but after that secondary treatment, only 2 out of 18, and once they were tertiary effluent, 0 out of 12. And so that's some good evidence that the wastewater discharged in the environment or reused may be safe and no longer infectious. So these are the couple of studies from Canada. So this one on the left here is from that Ottawa group I was just mentioning. They essentially were sampling mainly primary clarified sludge within or from the city of Ottawa, as well as the city of Gatineau. And they did a concentration step, and that is important to concentrate down the viral RNA if present. And then they did a nucleic acid extraction, so that's just extracting for the RNA. And then they did a reverse transcriptase, qPCR, to target regions known to be within the coronavirus. In this case, they looked at N1 and N2, which are very publicized by the CDC right now, so they're pretty good targets to choose. And as I mentioned, they were further able to show that when they saw more of a signal in the wastewater, proceeded 48 hours before an increase in clinical cases and 96 hours before those hospitalizations. So another group in Canada, so this is from the University of Dalhousie in Halifax, Nova Scotia. So they did a fairly similar kind of methodology. They got grab and composite samples from 3 communities in Atlantic Canada, and they underwent their RNA extraction and qPCR as well. Big difference here is that they didn't use a concentration step like the Ottawa group does and like a lot of groups do, and that has some benefits in that it saves a lot of money. It's a little more convenient, doesn't take as much time, and mainly it doesn't concentrate inhibitors. So if you have a lot of inhibitors in your wastewater, it can further affect your qPCR on the way down as well. And so they found similar results. They were able to detect some coronavirus RNA within the sewage as well. So I'll talk a little bit about what we have started to do at Queen's University. So we've joined the Wastewater Surveillance Initiative, which is supported by the Ontario Ministry of the Environment, Conservation and Parks. And so what we're doing is working with Utilities Kingston to get 24-hour composite samples 1 to 3 times per week. And that's from these 2 facilities, the Ravensview Wastewater Treatment Plant and the Cataraqui Bay one. And then those samples, they're either would be processed right away, which is our eventual goal, or they would be frozen at the appropriate temperature, depending on when we plan to process them. So our idea is that we would like to expand our sampling reach. So besides just looking at these 2 plants, we want to look at the Loyalist Township, which is quite close to Kingston, about 20-ish minutes away, for those who don't know, as well as the city of Cornwall, which is fairly closer to Ottawa. And then we're also interested in looking at specific community institutions. So things like prisons and post-secondary residences. We have a lot of them in Kingston. So we may as well take advantage of that and look at them. So this is the methodology in a nutshell that we're working on. So what's really important is that we're trying to get all of this to be able to do it in a 24-hour turnaround time. So right from sample collection to our qPCR results. That's really important because it's public health really needs that information as soon as possible if they're going to have some sort of a response to it in order to prevent an outbreak. So as you saw with that University of Ottawa group, they only predicted it 48 hours beforehand. So if we take 48 hours to process a sample, then that information is not that useful anymore. So I already talked about wastewater collection. We're getting those from Utilities Kingston. We did decide to do a viral concentration with a PEG precipitation step, and that's similar to what the University of Ottawa did. Then we're doing our RNA extraction and then our qPCR. And we're also targeting those N1 and N2 genes from the CDC, as well as an E gene, which is also often used in the literature. And we're also going to be targeting this PMMOV virus. And so that stands for pepper mild motovirus, and it acts as an endogenous fecal standard. So it's not part of coronavirus per se, like these three genes are. It actually just acts as a useful biomarker for the normalization of the signals of these three genes, because this pepper virus, it's found in all peppers. So if somebody is going to eat peppers, they're going to shed this in the feces, and we're going to be able to pick that up in the wastewater. So really quickly, just going over the status with Kingston and COVID-19, or more accurately, the KFLA region. And I think I got this, yeah, this dashboard is the latest one that came out. So we're relatively unimpacted compared to some other urban regions like Toronto, for instance. We're actually one of the 3 regions that were allowed to move back into a green status early on February 10th, I believe. So we've only had 707 cumulative cases and 1 death. And right now we're setting at 7 cases per 100,000 people. So because that case rate it's a little lower than some of those other urban centers, I was saying, we may not necessarily expect to see as high of a signal in wastewater as some of those other international studies. However, that's not to say that it's impossible. I did find this study from Japan where they had an even lower case rate, about 4.4 per 100,000. And when they had days or weeks where they were seeing higher caseloads, they were able to report more viral RNA in the population. That gives me hope, because there are definitely spikes in Kingston. We haven't been immune to that, even though our caseload has been relatively low. So of course, we had that first spike in wave 1 in that mid-March to April. We had a second one when we had a number of salons in the city implicated in having workers that tested positive. But that huge one is really what we would refer to as wave 2, and just this last late November and December, where there were multiple outbreaks at things like places of worship, car dealerships, etc. And so that really caused an increase of cases. So that gives us hope that we can pick up something in the sewage, hopefully before we see these peaks, and then appropriately influence public health strategies. And that's all. I just wanted to acknowledge my co-supervisors and then everybody else who has been working on this project with us. Thank you.
Thank you, Sophie. That was a great presentation. We do have a few minutes for questions. If anybody has any, please feel free. Here's one from Dr. Lougheed. Sophie, COVID-19 is contained in aerosolized droplets that are respired. We know that you can sample air for the signature of some organisms. Would it be fruitful to sample air in places that are frequented by the public, i.e. supermarkets, and try to test for different variants in real time? Assuming this works, perhaps using digital PCR, this approach might provide more spatially explicit insights on the presence of new variants. Would you like to comment on that, Sophie?
Yeah, I think that, yeah, it's certainly interesting, and I think that's something that could be possible, although maybe a little difficult, maybe more a little difficult than testing for sewage, but it has the same idea as testing for wastewater, because you're getting a bigger picture. You're not just testing one person, you're testing everyone who's been in the supermarket kind of thing. And it also has the potential to give you a bit more discrimination on places. So if you're testing the wastewater, you're just getting it from everywhere, and you can't say, oh, this is the one that came from this grocery store and this is what came from this university, unless you're going right into those facilities and testing it at a more upstream level. As in something like a supermarket, if you're going to test for droplets, then it's going to be right there, and you're going to know it was those people were at the supermarket. So I think that's interesting, and I'd like to look more into that. Yeah.
Okay. Thank you, Sophie. If anyone else has any other questions, please feel free to put them in the chat, and Sophie can answer you directly there. So we're going to move. Thanks, Sophie. We're going to move on to our last speaker, Corinna Dalistarna from the School of Environmental Studies. Her talk is titled, Can Canada's First Nations Water Security Be Engineered? The Role of Project and Engineering Management in Achieving Long-Term Infrastructure Service Delivery Objectives. Take it away, Corinna.
Give me a second to to just share my screen.
There we go. That looks good.
I'm just going to turn on my video as well. Okay. All right. Sorry, I think I just moved my slide forward. I'm going to have to do that manually. My keyboard all of a sudden is no longer working for me. All right. So good morning. My name is Corna Dalistrana. I'm a PhD student in the School of Environmental Studies. And I'm very happy to have been invited to speak here today. I'm supervised by Drs. Stephen Brown, Yves Fillion, and my advisor is Dr. Warren Mabey. So ensuring First Nations water security has been a challenge. And I will discuss why the role of engineering and project management is worthy of study and how it might help us get there, i.e. ensure water security for First Nations communities. Now, First Nations, let me start by saying that Maura Hanrahan indicated that Indigenous Canadians are 99% more likely to be water insecure than non-Indigenous Canadians. First Nations drinking water systems have unfortunately failed. They are underperforming, they're undersized, they're fragmented, and often use chlorination that leads to tap water avoidance. They're also not trusted, and that in turn too leads to risky consumption behavior. In 2011, a national study found that 73% of all drinking water systems, First Nations drinking water systems, were at medium or high risk of failure. Despite billions of dollars invested in First Nations water infrastructure, we are seeing them not performing the way they should. And so my project is trying to examine whether project management and engineering management might be holding some keys to the issue. Okay, all right. As of January 26 of this year, we still had 57 drinking water advisories in effect. Here, what we mean by that, long-term drinking water advisories have been in effect for more than 1 year. They caution residents to boil water, not to consume the water, or not to use it at all, even for bathing. The longest standing drinking water advisory has lasted for 26 years, but more to that later. The effects, of course, are multifaceted. The effects, of course, are multifaceted, and they range from spiritual to mental and physical health impacts to socioeconomics, impacts on the environment, and educational success, deterioration of housing, and the list really goes on. Most often or frequently, women and children are disproportionately affected. Okay, so something just, sorry about that. Sorry, when reviewing water security literature, there are some key elements and key factors that are affecting drinking water security. They range, of course, from droughts to floods, wildfires, natural disasters of any sort, and as well as temperature effects on water quality. We have physical determinants such as the geographic location, housing status, as well as the delivery of and storage of our drinking water, as well as our wastewater systems. Social determinants include, of course, socioeconomics, social structures, population distribution, and of course, also values and culture. The literature on First Nations drinking water and or Indigenous water governance is really rather rich. But what is apparent is that it is leaning toward the side of certain categories. There's a prevalent school of thought that holds that water crises are really a crisis of governance. And so we see categories of literature going into source control, a lot on values and ontology, and also on knowledge sharing and participation. What we see less of is what, again, the view holds, is that it is more a crisis of governance than of technological problems or even financial resources. We find also that the literature is fragmented. It is monodisciplinary. And what we are really lacking are multidisciplinary studies. And we are also seeing a paucity of engineering research. Now, let me explain why this might be an issue, that we have some problems here that we are not able to solve, and there might be information that is lacking. I will take you later to why I believe project management and engineering management studies could help us get there. So Neshkataga First Nation is a community of about 350 members that live on reserve. There's a fly-in community accessed in the wintertime by ice road. In the late 1980s, the community was dislocated and located to its current location due to infrastructure and flooding issues. In 1993, a new water treatment plant was built at the new location. Unfortunately, in 1995, shortly thereafter, the community went on a boil water advisory due to inadequate disinfection. This boil water advisory has not been lifted since. In 2017, the process for an upgraded water treatment plant was started. It was supposed to have been completed in 2018, but wasn't. Pump failures cut off water supply entirely in 2019, which led to an evacuation. A little bit more than a year later, another evacuation followed, this time because there was an oil sheen on the reservoir. After 2 months living in a Thunder Bay hotel, the Neshkataga people returned home. So although they have water now, their boil water advisory continues due to a number of different issues. And this, as I might want to point out, despite the price tag of currently $16 million. So I'd like to draw your attention to just a few issues that I picked up by surveying media reports. And I'm just going to talk about the first 4 highlighted in red. To start with, it is rather curious that now that the plant somewhat is completed, still has some issues, but now there's discussion of potentially having to replace the water distribution system, meaning the supply. What's also curious is that the advanced technology that has been used requires operator training at a Toronto level, highly sophisticated. Concern over the supply chain has also been voiced with respect to replacement parts and getting them to the community when needed. The overall system sustainability, of course, is directly and intimately linked to proper and timely management. So like me, you're probably wondering why, when we understand the system as it is, why is it that these factors were not considered at some point in time? The fact that the operator now needs to be trained by a professional operator who will have to be hired and live at the community for a year, and which is going to be financed by the federal government, tells us, in addition to all the other points that I mentioned, it actually calls into question that this community, even when the water treatment plant is going to be up and running and functioning fully, may not be water secure for quite a period. So what might be going wrong here? Well, researchers have indicated that and have observed that maybe the way we're managing our projects or even initiating and conceptualizing them is wrong. Currently, we're focusing on the project itself, the operational objectives of the project, and seeing the infrastructure project as an end in itself. We look at the life cycle as plan, build, maintain, and dispose. But what we should be doing is looking at the project as a system within a system. So the one asset and the project should be, its operational objectives should be matching with the system. The infrastructure project is really just a means to an end, and the end is to serve the community. But we don't necessarily see that happening. Projects, assets, and networks affect each other as they develop and age. So it's not static, but it should be a kinetic loop. So perhaps to illustrate the last point is that, as we know, chlorination as a disinfection method has certain quote unquote side effects, meaning we're looking at disinfection, chlorination byproducts, and of health concerns associated with those. In many other countries, these chlorination is no longer being used. So if we design a project or design a water treatment plan, for example, and not take into account that perhaps at some point we need to replace the disinfection system with something different and do that in a manner that causes the least disruption to the community, then maybe we are not going about the system or going about the process the right way. Now, even though what the First Nations communities are experiencing, what we see with their drinking water systems seems in Canada it's being focused on First Nations communities, of course. But what we are also seeing is that globally, there are many concerns about how infrastructure projects are underperforming, they're over budget, over time, and it has actually led Ben Fleiberg to refer to them as the survival of the unfittest. So in December of 2020, the Institution of Civil Engineers recognized that we need to do a better job. And it was prompted in part to something that the previous speaker was relating to or referring to, and that is COVID-19. We understand that we're expecting a huge influx of funds into infrastructure to build back after COVID, to help build back after COVID. So the Institution of Civil Engineers, recognizing these two things, essentially has concluded that we should be looking at a systems approach to infrastructure delivery, and we need to change engineering and project management. And we should keep the customer and operational needs at the heart of everything that we do. Now, this is going to be surprising to those who feel or who are familiar with human-centered design or even community-based approaches. But be that as it may, I was excited to find it because it actually allowed me to narrow down my research project. So my research question then is, what changes need to be made to Canada's First Nations drinking water infrastructure project and engineering management to achieve long-term operations and consumer benefits?
Karina, it's just 1 minute left.
Oh my gosh, I'm not sure how I did this. Okay, let me very briefly just point out that the life cycle of funded First Nations community infrastructure projects goes through 5 stages. But what we are not seeing is the feedback mechanism to identification of the community needs. In other words, how does the project in the end actually meet the community needs? I will be proceeding with a multi-case study using a multi-scale perspective by slicing it essentially vertically. I look primarily at planning approaches, engineering practices, and of course the technical artifacts, and use Bloom and Guthrie's strategic project lifecycle interfaces. They will allow me to look at feedback mechanisms as well as feed forward mechanisms to essentially give a coordinating structure to my research. I will draw on perspectives, cross-sectoral perspectives, ranging from community to educational institutions and government, and together, hopefully, arrive at findings that will help for transition to the factors that need to be incorporated into recommendations for long-term infrastructure service delivery. So key takeaways I have summarized here at the end. And in closing, I would just like to thank my supervisors and acknowledge also the various institutions from which I received funding. Thank you.
Well, thank you, Corinna. That was another great presentation. Thank you. That brings us to the end of the first session. I want to thank our 4 speakers. These were really interesting talks around some really critical issues with water security and equity and access. We're now on our break. So we have a 10-minute break. I would ask everyone to be back for 10 after 10 because we're going to get started sharp with my colleague, Dr. Brian Cumming, will be chairing the next session. Thank you, everyone.
Hello.
Hey, Shenyu.
Hi, Brian. How you doing?
Hello, how are you? I'm good, thanks.
Well, we supposed to be in the breakout rooms now?
No, I didn't. I think I hit a button by mistake.
That's what happened. I think I missed. I think I missed the breakout room invite.
No, there is no breakout room. If I hit something by mistake, then I don't know what happened there.
Yeah,
I was
Oh,
getting ready for the next session on water pollution and water quality research,
oh, okay.
which will be starting in 2 minutes. And our first speaker will be Shenyu.
Okay then. Well, thanks a lot.
No problem.
Hi, Brian.
Hi, Shenyu. How are you?
I'm good. How are you?
Good. And I'm assuming that you have access to share screen?
Yes, I do, I think.
Okay, excellent. And I'm looking forward to hearing your research.
Thanks.
Okay, welcome back, everyone. I'm Brian Cumming, and I'll be the moderator for this next session on water pollution and water quality research. I'm a professor and the head of biology here at Queen's, and my research is on aquatic ecology and long-term environmental change. We have 4 talks in this session, and the first talk is by Shinyu Sun and Shelley Arnott. Her talk is entitled Examining the Interactive Effects of Increased Salinity and Heat Wave on Freshwater Zooplankton Communities and the Influence of Timing. Shinyu, go ahead.
All right. Thank you. And hello. I'm Xinyu. I'm a PhD candidate in biology department, Queen's University. So today I'm presenting our research on interactive effects of increased salinity and heat wave on freshwater zooplankton communities. So I'll start with some background information. Freshwater lakes are important resources, but they are being disturbed by stressors. And one of them is increased salinity in lakes. For North America, the application of road salt is a major contributor, and the main component of road salt is sodium chloride. So this figure here shows a predicted chloride concentration of over 40,000 lakes in 17-state area in the US. And we can see that there are lots of lakes that are predicted to have a chloride concentration of higher than 50 milligram per liter, or even much higher. And why is that an issue? So according to a lab experiment conducted by Arnold et al. On Daphnia species, they found that chloride started to negatively affect Daphnia reproduction, even when it's still at a really low concentration. At community level, increased salinity can decrease zooplankton abundance, biomass, and richness, and eventually cause a shift in the community composition by eliminating sensitive individuals and species. Meanwhile, we're under the background of climate change. So climate change has caused various consequences, and one of them is increased frequency and intensity of heatwave events. This figure is part of the results of a prediction model. It showed how intensified lake heatwave will be under different climate change scenarios in the future. So far, only few studies focused on community level effects of heat waves in aquatic systems. However, those studies have shown that it can eliminate individuals and species that are vulnerable to such acute thermal stress and eventually change community composition. So the fluctuating salinity levels and increasing frequency of lake heat waves make high salinity and acute thermal stress likely to occur simultaneously, which means when the two stressors happen at the same time, or sequentially, which means when one stressor happens after the other. Despite the pressing issue of lake salinization and heat waves, little is known about how they interactively influence freshwater communities. So when we talk about combined effects of multiple stressors, we usually have three types of effects. So when the combined effects equals to the sum of the effects of individual stressors, we call it additive effect. When the combined effect is less than the sum of the effect of individual stressors, we call it antagonistic effect. And when it's greater than that, we call it synergistic effect. So there has been increasing interest in multiple stressor issues recently. However, past research has primarily focused on simultaneously occurring stressors. And very little has been done to understand how sequentially occurring multiple stressors affect communities in aquatic systems. And this has limited our ability to predict outcomes of future disturbances. To help fill this gap, we designed this study. So we aimed to answer mainly two research questions. One, is there any interactive effect of increased salinity and heat wave on freshwater zooplankton community? Second one is, how does the interaction between these two stressors differ when they are applied at the same time versus when they are decoupled in time? So we conducted a mesocosm experiment. We used zooplankton communities as our research objects. Zooplankton are an important component of lake food web. They connect the flow of energy and nutrients between primary producers and higher trophic levels. We divided our zooplankton communities into 4 taxonomic groups. We had immature and adult copepod. We have copepod napuli, which are early larva stage copepod. We had rotifers, which are really tiny zooplankton. We also had cladosmer. We conducted our experiments. We conducted our experiments at Queen's University Biological Station, the star there. And we collected zooplankton from 6 different lakes around cubes and mixed them to create a diverse zooplankton community. In this picture, these are my mesocosms. So we had 6 treatments in the experiment. We had a salt plus heat treatment for sequential scenario, where communities were exposed to salt for 8 weeks, and then salt was removed and they were heated for 3 days. We had salt plus heat treatment for simultaneous scenario, where communities were exposed to salt for 8 weeks, and during the last 3 days of the exposure, they were heated at the same time. We had a heat only treatment for both scenarios. We had a salt single treatment for simultaneous scenario, and we had a salt single treatment for sequential scenario, and we had a completely controlled group. we got some really interesting results. So first, we found salt by heat antagonistic effects on total zooplankton abundance and biomass. We didn't find any interactive effects on total zooplankton richness. So it's antagonistic effects. It means that the combined effects were less than the sum of the effects of the individual stressors. Because our total abundance and biomass data share similar patterns, so here I only put total zooplankton biomass plots here for simultaneous and sequential scenario. We can see that the combined effects, which are the last boxes, the effects of the combined treatment were similar with the salt-only treatment and stronger than the heat treatment. We also found similar patterns for simultaneous and sequential scenario. We can also see that from these figures. These two plots have, in general, similar patterns of the effects. So these are box plots of biomass of each taxonomic group for simultaneous scenario. We didn't find any interactive effects on any of the taxonomic groups. But if we look at each taxonomic group, we can see that neither stressors affected copepod biomass, significantly affected copepod biomass. But our heat treatment negatively affected Napuli, rotifer, and Cladoceran biomass. Our salt treatment negatively affected Napuli and Cladoceran biomass. It didn't significantly influence rotifer biomass. We have similar patterns for sequential scenario, just slightly different. Again, the salt treatment negatively affected Napuli and Cladoceran biomass, but this time the rotifer biomass in the salt treatment is significantly higher than that in the control group. So, and we also, for each scenario, we also conducted an indicator species analysis, which allows us to identify species that represent each community. So this is a PCA plot of indicator species for simultaneous scenario. And we can see that our control group and heat treatment shared similar indicator species, while our salt and salt plus heat treatment shared similar community composition. And the salt treatment caused a significant shift in the community composition. Similarly for the sequential scenario, again, the control and heat treatment share similar community composition, while the salt and salt plus heat treatment share similar indicator species. And salt caused a shift in the community composition. They're in the nearly opposite direction. So now if we go back to our original research questions for the first one, the interactive effects between the two stressors. We think that it's possible that the heat effects were masked by the impacts caused by the previous salt exposure, which caused such antagonistic effect. We think that it could be that the sensitive individuals have already been eliminated by the previous salt treatment. And our heat treatment was not strong enough to cause further influence on the individuals that survived the previous salt exposure. So that's why we could see that our combined treatment had similar effects with the salt-only treatment. And for our second question, we didn't find different patterns between the two scenarios. And actually, before the experiments, we were expecting that when the two stressors were decoupled in time, the individuals will start to recover when the first stressor was removed. So the effect will be different from our simultaneous scenario. However, we didn't. And it's possible that although the two stressors were not applied at the same time in sequential scenario, heating was started immediately after the removal of the salt treatment. So we didn't really give the community enough recovery time, and there was legacy effect from the previous salt exposure. So some main conclusions of the experiment. So we found salt by heat antagonistic effects on total zooplankton abundance and biomass in both simultaneous and sequential scenarios. Even though the two stressors were decoupled in time, there could still be interaction between them. And it's possible that when there are different time intervals between them, which means when we give them different recovery times, the effects will also change accordingly. But that's kind of like our next research direction. So acute thermal stress has been infrequently considered in multiple stressor studies, particularly for community level experiments. However, our study suggested that short-term exposure to heat could cause community level damages by reducing zooplankton abundance and biomass. Therefore, we think that more attention should also be given to such emerging issue. And that's it. Thank you.
Okay, we have 2 minutes for questions. If there are any, please put them in the chat. And we have one from Steve Lougheed. Nice to see your cubes research featured. In your ordination plots, the space occupied by your control replicates was markedly smaller than in any of your treatments. Does this speak to the idiosyncratic individual responses of your indicator species to different factors, or to some other perhaps uncontrolled environmental factor?
Yeah, thank you. It's a good question. So that's something we are, to be honest, not really sure about. We think, because we did find previous research saying that by adding the stresses in, it actually caused more like variation in the community, and we think it's possible that it's due to the the variation of the variation in the sorry. So it's could be that the variation of the individual responses to the stressor. So we know that even within a single population, different individuals could potentially respond to the same stressor in slightly different way. So that's a potential explanation of that variation in that. And because for the control group, there was no stressor in it, so we didn't see such variation. But when there is stressor added, we see that such variation. Yeah.
Okay, thank you very much, Shinyo. Very nice. Our next speaker in the session is Alan Tien, who's a PhD student with Yusheng Wang in the Department of Biology at Queen's. And he's going to talk about early detection of toxic algal blooms using unmanned aerial vehicles.
Thanks. Hi, everyone. I'm Alan. I'm a finishing master's and starting PhD student in Dr. Yusheng Wang and Dr. Daniel Lefebvre's labs at Queen's in the Department of Biology. And today I'll be talking about detecting algal blooms using UAV. So first off, what are algal blooms? So cyanobacterial harmful algal blooms, also known as blue-green algae, are an increasingly important issue in the degradation of aquatic ecosystems. They consist of dense surface aggregations of cyanobacterial species, and they can produce cyanotoxins, block sunlight from other plants, create large noxious dead zones when they decay. And this can create fish kill zones, disrupt aquatic plants, reduce water quality in general, impact human health and recreation, disrupt drinking water sources. And their prevalence and impact has been increasing in the past 4 decades. And promoting factors include human nutrient loading, increases in solar radiation, and water/sediment temperature from global warming, ice cover disruption, internal nutrient loading from sediment from boating activity, and disruptions in wetlands and aquatic plants. So an important life cycle characteristic to note of several cyanobacterial species is their buoyancy control. Through inflating or deflating gas vesicles, cyanobacteria can move up and down in the water column, and this can occur both in their annual overwintering cycles and in daily cycles. So cyanobacterial species such as Microcystis typically overwinter in the bottom of the water column, and spring recruitment is thought to be triggered by rising temperature, solar radiation, which indicates the disappearance of ice cover, and turbulence. They then enter a growth phase where individual cells reproduce. And then a lot of species can enter a colonial phase where cells form tight groups bound by extracellular polysaccharides, which is a slimy coating akin to a biofilm. And this is what we typically see in algal blooms. And in the autumn, surface blooms sink and are thought to overwinter at the bottom of the water column. On a daily scale, cyanobacterial species such as Microcystis control their buoyancy through their gas vesicles and through producing producing carbohydrate ballast. They fill their gas vesicles through respiration, primarily at night, and this increases their buoyancy. And they float to the surface by early morning. Photosynthetic activity then reduces the size of gas vesicles and produces carbohydrates as ballast, and then they sink in the water column in the afternoon. And this is a fairly slow process, so they usually sink and rise once or twice a day. But this is also mediated by colony size. So colony structures enhance buoyancy control with an optimal size between 160 to 400 microns, and buoyancy and position is further mediated by abiotic processes such as precipitation and wind. So strong wind or rain can mix the water column and sink the cyanobacteria. And because of this, blooms can disappear and appear in a matter of hours, and tracking small blooms that are affected by weather is very difficult with current methods. So here's some more drone photos of blooms. And so traditional remote sensing methods are inadequate for monitoring blooms in their early stages or in smaller lakes because of how transient they are. So in this figure, green represents economic efficiency, pink represents detection ability, and blue represents frequency. The y-axis is temporal sampling frequency, potential of different data collection methods, and the y-axis is spatial coverage. So low resolution satellites are low cost for collecting data, but they lack detection ability. High resolution satellites and small planes or manned aircraft have high detection ability, but can't be deployed on demand. They cover areas infrequently and they can be very expensive to collect data from. Ground surveys are limited by accessibility and lack coverage, and sensor networks are very expensive and lack coverage as well. So there's a bit of a gap in detection ability. So we decided to tackle this project with UAV imaging, which can be deployed on demand, is low cost, doesn't require much training and permits, and has very high resolution. And through using a consumer RGB camera instead of spectral sensors, combined with a detection algorithm, we can reduce payload and cost while retaining sufficient detection ability for early monitoring. So here are some of the benefits of drone imaging. The image on the right is one that we took in China in 2018. So we can obtain data on demand. It's really rapid, and processing can be conducted on site in less than an hour. We can adjust the resolution and coverage, and it's very low cost. And with a two-step RGB algorithmic detection workflow, we reduce the need for expensive spectral imaging. So using this, we aim to answer some research questions. What are the main local abiotic and biotic environmental and geographic factors that drive the distribution and coverage of small chabs? So we hypothesize that emergent macrophyte coverage, high offshore wind speed and precipitation will inhibit chabs, while high water and air temperature, pH, and solar radiation will promote chab coverage. And what timing of these factors are the most significant? And so we hypothesize that macrophyte coverage early in the year will inhibit chab coverage, reducing benthic recruitment and early growth. Cumulative precipitation over several days will mix the water column and reduce chab coverage. And previous day light and humidity and same day wind will also be strong predictors. So to do this, we conducted drone flights. So we did 50 drone flights using a Mavic Pro drone, which is a consumer grade UAV with a 12 megapixel camera. Our imaging covered a 30-hectare area with a resolution of 3.1 centimeters per pixel and 25-minute flights. Surveys were once every 2 weeks in June and July, weekly in August, and every 3 days in September and October. And this was based off of how likely chabs were to appear. We also collected water temperature, pH, and connectivity on site during the drone flight. And we binned our weather data into 1 hour, 12 hour, 24 hour, previous day, 2 day, 3 day, 5 day, 7 day averages or sums, or in case of precipitation. And we also binned wind into offshore and inland directions. And this was our sampling site. So we decided to work on Dog Lake, the 5th last lake in the southern end of the Rideau Canal. And this was an ideal site for many reasons. It has a history of annual algal blooms. It has 2 deep natural basins and a 3rd newer artificial one that was formed after the construction of the Rideau Canal. There's a public beach, and which makes access easier, especially for a eDNA study we conducted as well. And there's historic background data on total phosphorus, calcium, and SETI depth from the Lake Partners Program, and also historic testing for cyanotoxins. And the southern bay, which is where we conducted our drone study, has a history of severe blooms. It's called Milburn Bay. So we divided our study of Milburn Bay into 300 meter by 100 meters sampling quadrats, and they were the closest areas to our sampling sites for water quality parameters, temperature. So this was our UAV workflow. Before the flight, we would check the weather. If it was too windy or rainy, drone flights wouldn't be possible. And we planned our flights. We checked our cameras. On the day of, we set up reference boards for normalization, and then we conducted a flight. And then afterwards, we did radiometric correction and geometric correction. We aligned the images and then used photogrammetry to stitch the images together. We applied our detection algorithm, and then we analyzed coverage with ImageJ. So this was how we developed our enhancement algorithm. A previous PhD student in our lab examined the spectral reflectance peaks of cyanobacteria and mapped these peaks under RGB space and created an algorithm for enhancing reflectance peaks and reducing pixel brightness of other signals. So this algorithm uses forward principles components rotation and k-means clustering. The user selects what classes are what. So essentially, the image is divided into a set number of color clusters, and then the image selects which ones are cyanobacteria, which ones are macrophytes, and which ones are background. And previous grad students use the confusion matrix for validation. And then we used ImageJ with the yen thresholding settings for area quantification. So this is an example of our algorithm at work. So this is an image taken with a UAV. And then these are the image clusters. And then after user selection, the background is darkened and the macrophytes are also darkened, while the cyanobacterial signal is brightened. And then this is applied back to the original image. And then using ImageJ, we can quantify the area. And then we plugged our predictor variables into SPSS's automatic linear modeling function, and we cycled our different predictor variable timings through and to find the ones with the lowest information criterion value. And then we used best subsets to find the strongest predictors. And we ended up with a model with the average depth of each site, the average air temperature of the previous day, the water temperature, 3-day cumulative precipitation, the average solar radiation 1 hour prior to the flight, and average plant area 2 months prior to the flight in our model. We also examined the impact of wind direction. So for that, we just did simple t-tests. So the 2 graphs on the left are cyanobacteria, and the 2 graphs on the right are macrophytes. No significant effect was found for macrophytes, while wind from inland direction, so if it came from the direction of the tree cover, had a significant impact. And chab coverage and particle size were higher when wind was inland. There was also a significant correlation between wind speed and particle size. And as wind speed increased, particle size decreased. So in summary, we successfully detected and quantified some small chabs from their onset to their disappearance over a 5-month field season. We created a regression model incorporating abiotic and biotic predictors with an R-square value of 0.52. We found positive correlations for air temperature, precipitation, and negative coefficients for solar radiation, water temperature, depth, and plant area. Solar radiation and water temperature were very interesting for negative correlations. And for water temperature, this was likely because of the way we measured it. So we did a one-time measurement in the early morning when we conducted our flight. And this was likely when water temperature was at its lowest. And so chabs are promoted over other phytoplankton by high temperatures, but they're also promoted by high temperature flux. So this low water temperature at this one time of the day probably indicates higher daily temperature flux. Solar radiation might have been because of greater photosynthetic activity in the morning, which would have increased their carbohydrate ballast and decreased buoyancy. So that was probably also a timing effect. So timing and duration of environmental predictors are obviously very important. And we also found that collinearity is a really big issue with environmental predictors. And overall, UAV are very powerful tools for ecological monitoring. And that concludes my presentation.
Thank you, Alan. We have time probably for one question if someone wants one in the chat. Okay, I don't see anyone coming up with questions, but I can ask a very quick one. When you're trying to estimate, sorry, you detected around 50% of the variation in your coverage of your chabs.
Yeah.
That's quite good. And you mentioned about collinearity. What can you expand on that just a little bit?
So we had to exclude factors like humidity because it had such high collinearity with things like precipitation.
Okay. Well, thank you very much. I'd like to thank Alan for his talk, and we'll move on to our next talk. It's just around 10:40. The next talk is by Catherine Moyer, and it's a collaboration between Matt Windle and Jeff Riddell at the St. Lawrence River Institute, and Zoe Armstrong and myself at Queen's University. And Catherine will be talking on lower trophic level responses to legacy sedimentary mercury contamination on the St. Lawrence. And go ahead, Catherine.
Perfect. Thanks, Brian. I assume you can see that. Okay, so yeah, as Brian said, this is a collaborative study, a couple of studies really, between our lab at Queen's, which is the Paleo-Environmental, oh God, I'm not even gonna say it, Paleo-Ecological Environmental Assessment and Research Lab, or PEARL, and the River Institute in Cornwall. So thankfully for me, oh no, are you not gonna work? Come on, little guy, there we go. Yeah, thankfully for me, Jeff gave a pretty thorough introduction of the Cornwall area yesterday in his plenary. But as he mentioned, it's an area that's seen quite a lot of industrial history throughout much of the 20th century. And a lot of this activity was really situated right along the waterfront. So there was a lot of effluent being delivered directly to the river through much of the 1900s. Oh, come on. Sorry, this is really slow. Yeah, this is a figure that Jeff presented yesterday showing essentially surface sediment concentrations of mercury in the St. Lawrence River at Cornwall and then downstream in Lake St. Francis. So prior to about the 1970s, there weren't a lot of regulations in terms of what could be loaded into the river. Concurrently, in the 1970s, these laws got enacted, a bunch of improvements to industrial processes occurred, and so we saw a drastic decline in how much mercury and other metals were being loaded to the river. So as we can see on the right, kind of a steady decline in sedimentary mercury concentrations downstream of Cornwall. We can also see this in aerial imagery. I don't have cool drone shots like Alan does, but I do have historical air photos. So the upper image here from 1972, you can actually see the plume of effluent coming out. There was quite a lot of industry located kind of just up here, and it was released into the river. You can see it coming out, hugging kind of the North Shore. And once these kind of improvements were put into place, you can no longer see this plume coming out. And as Jeff mentioned, primarily this has been important for restrictions on fish consumption because the actual mercury concentration in fish is still quite elevated in the area. But if we zoom in on the Cornwall waterfront, we still have some regions where mercury concentrations remain elevated in the sediments. So notably, there have been kind of these 3 zones along the Cornwall waterfront that have been delineated where elevated sedimentary mercury concentrations remain an issue. So these data on the right are from a recent study that we completed looking at surface sediments from very nearshore sites in these 3 zones. And what we can see is that in all of these 3 zones, we're elevated in both total mercury and methyl mercury concentrations relative to upstream reference sites. So we know that our sediments are elevated in mercury here, and we know that there have been some evidence of fish being elevated in mercury and that being a concern. But what about other organisms, particularly those that are living in or on the sediment that are potentially exposed to this high mercury? So what do we know about how metals affect these organisms? Well, we know quite a lot about other metals other than mercury, particularly those that are present in mine waste commonly. So these are pretty well studied. We know a little bit about waterborne mercury, but there haven't been really a lot of field studies looking at sedimentary mercury impacts on benthic dwelling organisms, particularly at the concentrations that we see. The few studies that I've found, the highest concentrations of mercury that they have tend to be kind of like 1 to 2 orders of magnitude lower than what we're seeing. So it's important to understand how the biota are reacting in this natural setting. As well, Zoe, who Brian mentioned, is another PhD student in our lab, wanted to do a kind of finer taxonomic resolution on the chironomids. There's been a little bit of research on chironomids in the area, but she wanted to narrow this down from kind of a family level to genus and species if possible. So that brings us to the questions we were trying to address, which is really, do chironomids and benthic diatoms respond to elevated sedimentary metal concentrations, particularly of mercury, because it's such a concern in the area? And overall, has community structure changed in response to remediation over the last 10 to 15 years? Okay, so if you're not familiar with chironomids, I keep using this term, you're like, what is she talking about? These are non-biting midges. So they're little insects, and they spend their larval stages living in the benthos before emerging as flying adults. So importantly for us, they produce these chitinous head capsules, which tend to preserve well in sediments. So this is just a photograph taken through a microscope of what one of these preserved head capsules look like. And these capsules have different features that you can use to identify the taxon that you're looking at. So things like the mentum, which is like the teeth here, the mandibles, the ventromedial plate, and other features. So you can use that to identify your typically genus. The other group that we're looking at are the diatoms. So these are single-celled algae that also preserve very well in sediments because they produce this siliceous cell wall. So it doesn't break down like organic material is going to. And importantly for diatoms, chironomids tend to respond very well to pollution. Diatoms also respond to various environmental factors, things like nutrient concentrations and pH, as well as pollution. And so this type of kind of relationship really forms the basis for what our lab does, which is paleolimnology. So it's looking at things preserved in the sediments, in this case, biological communities, and using that to reconstruct past environmental conditions. So in terms of what we actually did, we had surface sediments collected from nearshore areas in our 3 contaminated zones. These green triangles just kind of indicate where major historical industrial sources would have been, as well as some upstream reference areas. And we collected the top 2 centimeters of sediment. So we would just extrude the top 2 centimeters, did a whole suite of elemental analyses and metal analyses on them, as well as grain size, total organic carbon. And for all of these sites, we subsampled for diatoms. And then for a subset of the sites, we subsampled for chironomids as well. So we have our surface sediments. We also took a sediment core from one of the zones, which is indicated by this blue square here in zone 1. And with sediment cores, essentially, this is what's going to allow you to construct a ecological history of the area. So the idea is that recently deposited material is going to get overlain on top of older material, assuming that your sediment's not disturbed. So then you can slice up this core into little segments and use that to create essentially a history of your area. Okay, so in terms of the actual concentrations of metals that we found, I'm going to be presenting this as PECQ values. So what this is, the PEC is a probable effect concentration. So that's just, it's like a sediment quality guideline. It's a concentration above which adverse effects are likely to occur. So for each metal or metalloid, this number is going to be different depending on its toxicity. And then the PECQ is just the concentration in your sediment divided by that guideline. And really what this allows us to do is to make it easy to compare between elements. And so anything with a value over 1 is going to be likely to have adverse effects for sediment dwelling organisms. So what we actually see in terms of our results are mean PECQ, which is an average across all of the contaminants of concern that we actually had PEC values for. We have, for the most part, below 1 across, if you average across all of your elements. But we do have a couple of trends and differences here. Notably, zones 1 and 2 are elevated, in our mean, in our total mercury, as well as in our lead, and then zone 2 in our zinc relative to our reference sites. And then zone 3 is lower, but also still elevated relative to our reference conditions. So we do see sedimentary contamination in these sites. But again, to note, most of our values are actually below this threshold value of 1, with the exception of mercury, which tends to be quite elevated. Okay, so for our surface sediments, our diatoms, what we did was essentially a kind of complicated spatial analysis that I'm not going to go into here. But what we ended up with as a result of this analysis was a number of spatial predictor variables that we could then put into an ordination. So we did redundancy analyses with our environmental variables, so our metals, our TOC, our grain size, depth, things like that. And then we did another one with our spatial variables. And what we found was that they were both important factors in influencing our diatom assemblage structure. If we look at our triplot here on the right, we see a couple different things. We tended to have our industrial contaminants co-vary in kind of the same direction. So zinc, lead, copper, and mercury in particular. And then we saw most of our reference sites were clustered in kind of the opposite direction of that, which is really what we'd expect to see. We did have some diatom species that tended to co-vary with industrial contaminants, as well as some that were inversely related to contamination. But this has been a little bit inconsistent with previous research. Some of these individual taxa that ended up being really metal sensitive have been previously reported to be metal tolerant. So not too sure what's going on there. And we didn't have any real indicator taxa that were present in super high abundances, say in highly contaminated sites and not at all in others. Really what we saw is that it was slight shifts in community assemblage structure. We also did a variance partitioning analysis using our environmental and our spatial predictor variables. And what we saw was that they both contributed significant amounts to the variation in our diatom assemblages, but relatively small amounts, both about 14% individually. And important to note here is that our residual unexplained variation was quite high. So probably a lot of variables that we didn't measure that are importantly influencing our diatom community structure. For the chironomids, again, this is Zoe Armstrong's work. One of the main things we found is that chironomid diversity wasn't impacted by contamination, which is kind of what the opposite of what we expect to see. Normally, the more contaminated your site for chironomids, the lower you expect your diversity to be. We did see some assemblage structuring, but it was mostly to do with zinc, copper, and lead, which are all kind of highly correlated, rather than with mercury. So we had some degradation tolerant taxa that increased in abundance as our zinc and other correlated metals increased. We also had some generalists that responded the same way, as well as a few metal sensitive taxa that declined as our concentrations of metals increased. For our sediment core, if you haven't seen a stratigraphy before, we use these a lot in paleolimnology. Essentially, our independent variable here is our depth, and then we have dated our core, so we can apply dates as we go down core. And then we have our individual taxa listed here, as well as our total mercury on the right. And really what we see here is a huge shift from kind of prior to the 1960s, from really planktonic taxa, to mid-1970s, a shift to smaller benthic taxa. And interestingly, what we've noticed is that a lot of these planktonic taxa are really eutrophic species with high nutrient requirements. So our thought for this is that, yeah, we had high mercury at this time, but we also had extremely high organic loads coming from upstream. So it was a pulp and paper mill that was operating just upstream of our site. So there was mercury and other things coming in, but really what we think was structuring the diatoms was these huge amounts of nutrients being loaded in. And if we look at an aerial image, this is our coring site in red, and you can actually see the plume of effluent coming over. So we think that this is probably due to these high organic loads of nutrients coming in. But it's hard to disentangle because kind of all of these industrial improvements came in at the same time. So reductions in mercury and nutrient loads were happening at the same time. For our chironomids, really the most interesting thing to note here is that we had hardly any chironomids prior to about the 1960s. In more recent terms, we saw a bunch of degradation tolerant taxa occurring, but really this extremely low concentration is quite unusual. And again, what we think is happening, our best guess anyway at this point, is that it's possible that these really, really high BOD loads, so a lot of oxygen is going to be needed to break down all this organic material. Our best thought is that maybe there wasn't actually enough oxygen there for chironomids to survive. So just to conclude, sedimentary contamination, particularly of mercury, but also some of the other metals, has a small but significant impact on our benthic diatom assemblages. Although we think that the natural water chemistry of the St. Lawrence River, which has, a bit slightly alkaline pH, relatively hard water, could be providing some protective effects from a kind of toxicological standpoint. Unfortunately, our sediment core resolution was too coarse to infer recent remediation impacts in kind of the last 10 or 15 years. We also didn't get back far enough to see pre-industrial conditions. So I know that Zoe wants to go back and try to get a longer core and section it more finely to be able to infer more recent impacts as well as comparisons to pre-industrial times. We think that generally these historical changes were probably driven more by nutrient loads than by metals. And so generally, these metal concentrations are important and we should consider them as factors in contaminated areas. But if you hear hoofbeats, don't think zebras. So there may be more obvious explanations like nutrients coming into play. And that is it for me. Thank you very much.
Thank you, Catherine. And that was very interesting. If anyone has any questions, please put them in the chat. We're out of time and we're going to move on to the next talk, which is by Dexter Dean Cockerone, basically at the University of West Indies. And his presentation is with Gail Prasad Ford and Mona Weber. And the title of the talk is An Assessment of Water Quality and Plankton in Key Areas of the East Portland Special Fisheries Conservation Area, Jamaica. Go ahead, Dexter D.
Thank you, Dr. Cumming. I was just trying to share my screen here. Just give me a second here. Not allowing me to share here.
You can share, Dexter Dean, is that the problem?
Yeah, I have a problem sharing my screen here.
So when you hit the green button on the bottom that says share screen, it doesn't work?
No, my list of applications come up. I click on it and it's saying it cannot grant access.
Do you know my email? If you can email your presentation to me, then I will open it up here locally. It's just L-O, I'm giving this to everybody here, excellent. L-O-U-G-H at Queensu, Q-U-E-E-N-S-U.ca.
Yeah, just one more time for me.
Yep, it's L-O-U-G-H at queensu.ca, Q-U-E-E-N-S-U.ca.
All right, attaching. Sorry for the delay, everybody.
So did you manage to send it, Dexter Dean?
It's attaching.
Okay.
Attaching. Sorry for the delay, everybody.
Yeah, don't worry about it. Stuff happens. We all do field work.
Yeah. It's about halfway there.
We'll get it. People can go make themselves a tea, do something good.
Trying to share my screen again.
Yeah, it's shared.
Okay, nice. Great.
You're live. Very
All
good.
Right. All right, again, sorry for the delay. Good morning still. All right, my name is Dexter Dean Cahoon. As you heard, I'm a PhD student in the Department of Life Sciences at the University of the West Indies, where I'm supervised by Professor Mona Weber, and my advisors include Dr. Dale Prasad as well as Dr. Weber, Professor Dale Weber. So the title of my presentation today is an assessment of water quality and plankton in key areas of the East Portland Special Fishery Conservation Area. And this is essentially chapter 3 of my PhD thesis, which is entitled an assessment of water quality, plankton, and trophic status towards creation and management of a special fishery conservation area in East Portland. So my presentation will follow the outline shown here. And I'll begin straight away with a brief intro and background. So special fishery conservation areas, what are these? These are no fishing zones or areas reserved for the reproduction of fish population. And specifically with regard to Jamaica, the nature reserve status is declared under Section 18 of the Fishing Industry Act, and that makes it illegal and punishable by law to engage in any unauthorized fishing in these areas. So just for a quick clarification, special fishery conservation areas were previously called fish sanctuaries in Jamaica, and they still are commonly called fish sanctuaries, but they were actually later renamed to allow some flexibility in fishing. That's quote unquote necessary fishing, which was allowed, such as for conservation purposes, the removal of invasive species such as lionfish, for scientific assessments and so on. But underlying principle is the same for both, and in fact, I may use both names interchangeably throughout this presentation. So the value of fish sanctuaries are the same, they are widely accepted as effective, inexpensive fishery management tools, but their value extends much further into other areas such as economically, recreationally, educationally, and more. And in Jamaica, the selection criteria for these areas or potential areas is very robust. Is a very specific selection criteria proposed by Aiken et al. In 2011. And they include aspects such as the area must possess healthy undamaged coral reefs, moderately large numbers of reef fish and associated species, as well as adjournment with adjoining integrated ecosystems such as mangroves and seagrass, and a list of other social and scientific considerations. But the scientific evaluation is an important criteria in the selection process. Let's just get that out there. But perhaps what's missing from the list of criteria is the use of ecological indices such as planktonic and physical chemical data, which are commonly used to indicate ecosystem state or health. These indices may be equally important or equally as important in identifying such areas or on the other hand, enriched or eutrophic areas. So a part of my overall research included using these indices to select the ideal area in East Portland for our fish sanctuary, whether it was a continuous area or a discrete area, as well as to determine the trophic status. But these indices have much further uses than that. They can also, as I mentioned before, indicate areas of concern, which is the main focus on this presentation today. So the planktonic indices or the ecological indices, they include both planktonic and physical chemical parameters. For example, zooplankton assessments, which would include total abundances as well as composition or distribution of key taxonomic groups. Phytoplankton assessments, that would be with regard to total and size fractionated. As we may know or may not know, the determination of phytoplankton as chlorophyll A in size fractions can be extremely useful, as there's a direct relation between the phytoplankton size and nutrient concentrations, where we normally find the bigger size fractions in the more eutrophic or enriched areas, versus the smaller size fractions would mostly be associated with more oligotrophic or pristine conditions. So nutrient analyses involve the use of hydrographic data such as temperature, salinity, TDS, pH, and so on. And these in tropical environments can be especially useful for indicating riverine inflows or eutrophic periods. Similarly, nutrients such as nitrates and phosphates have been used to confirm events of eutrophication or nutrient upwelling in areas. And these of course would be residual nutrients in the water column. But the underlying principle is that increased planktonic productivity is usually associated or attributed to influxes in river-borne nutrients following rain in the tropics. So the general aim today is to compare the planktonic and physicochemical indices at the specific enriched areas in the East Portland Special Fishery Conservation Area and suggest possible reasons for the water quality conditions in these areas. And the secondary aims seek to determine which indices are useful for continued monitoring of these areas, as well as which station or stations may appear to be most exposed to enrichment. So the specific research questions are, what, if any, are the likely sources of anthropogenic stress contributing to deleterious water quality conditions in the East Portland Special Fishery Conservation Area? 2, are there any differences or similarities between the biotic and abiotic indices at the indicated areas? The third question is, are some indices more useful for continued monitoring of the area? And the fourth question is, what station or stations appear most exposed to enrichment? So for those not familiar with Portland, Portland is Jamaica's northeasternmost parish. And it's particularly unique because of features such as its dynamic topography. It has the highest annual rainfall on the island, between parishes and the island. And it has generally a very high biodiversity, but much of it is unexplored. But there was a great paucity of data, especially scientific data, marine scientific data for Portland prior to the initiation of this research in 2014. So as I mentioned earlier, a part of my research helped with the designation of the East Portland Special Fishery Conservation Area. This was declared in July of 2016. And the areas we're looking at today within the sanctuary include Turtle Crawl, indicated by the red arrows there, Turtle Crawl, Sansan Bay, West Channel, and the Blue Lagoon. So in terms of the methodology, the field methods include physical chemical sampling using a multiparameter. The phytoplankton samples were collected using a 5-liter Niskin bottle, and this was done just below the water surface. Zooplankton samples were collected using a 335 micron plankton net, and this was used in conjunction with a flow meter. Those were done for 10 minutes in wide circles at approximately 0.5 meters per second. So in lab, the zooplankton were sorted, total abundances were determined, as well as abundances of the specific taxonomic groups. This was done using a microscope, of course. Phytoplankton were filtered using Nalgene size fractionating towers. Following that, extraction was done using 90% of 6 mils of 90% acetone, and the aliquot of that was then put in the fluorometer after 24 hours. In terms of the physical, chemical, and nutrient data, these were the nutrients were determined from the filtered seawater from the phytoplankton analyses. Those were sent off to a lab for analysis for nitrates and phosphates. And the physical chemical data were sorted and assessed for significant differences between stations using Statistica. So I look at the results. With regard to the physical chemical data, only 3 of the 7 parameters indicated significant differences between stations. These were salinity, TDS, and conductivity. With regard to nutrients, the nitrate levels were significantly different between stations. However, phosphate levels were not deemed significantly different between stations. It was noteworthy to mention that stations such as West Channel and Sansan had relatively high nitrate levels or concentrations, while stations such as Turtle Crawl and West Channel again had relatively high phosphate levels. With regard to the phytoplankton analysis, none of the parameters indicated significant differences between stations, but graphs were still plotted. And from these graphs, you could actually see some differences graphically between stations, such as high absolute abundances at station 1 and 11, that's Turtle Crawl and Blue Lagoon, respectively. Those are both inner stations. while generally outer stations such as Station 2, Station 7, those generally had low chlorophyll A or phytoplankton biomass. With regard to the zooplankton, most of the parameters, the zooplankton parameters indicated significant differences between the stations. The only parameter that did not indicate significant difference was the group Larvaceae, and that showed no visible trends. And with regard to the total zooplankton abundances, the total abundances were dominated by the herbivorous calanoid copepods at most stations. Turtles such as Blue Lagoon and turtle crawl again had high zooplankton abundances, and Sansan had the least abundances. With regard to the composition, on the left there, that's the herbivores, the main herbivores, which are the calanoid copepods, the dominant community. In this investigation, you can see that numbers were highest at inner stations again, such as the turtle crawl and Blue Lagoon. And the graphs on the right in red are the carnivores. And you can see a clear antagonistic relationship, such as at the outer stations, the carnivores are more dominant versus the herbivores being more dominant at the inner stations. So I look at the discussion. The discussion will be, the results are discussed with regards to the research questions, of course. And the first question was the likely sources of, was about the likely sources of anthropogenic stress. There was unexpectedly high phosphate levels at the outer station at Turtle Crawl. And this was, likely attributed to an adjacent hotel nearby. Likewise, high nitrate levels and phosphate levels, as well as low TDS and high salinity was observed at West Channel. And that particular station was located just offshore a very populated coastline. So we attributed the very high nitrate levels there, up to 8 times higher than the other stations, attributed those levels to possibly leaching from underground or release from underground. Because prior to the development of the sewage plant in Portland, there was widespread use of soakaway pits and latrines and pit latrines in the Portland area. With regard to the similarities or differences between the indices, the 3 parameters that indicated significant differences were useful indicators of the presence and influence of riverine inputs in the bays, and even the fact that most of the parameters were not significant. The physical chemical parameters were not significant different, that also attested to the general uniformity or homogeneity of the East Portland area. Another similarity was that the inside bay stations were dominated by herbivores, as I mentioned before, while the outside bay stations had more diversity and more carnivore groups. And the only difference was Sansan Bay, which was an inner station that had low total zooplankton abundances as well as diversity, but also noticeably there was a high net plankton size fraction proportion, which was altogether, these were indicative of eutrophic conditions. So with regard to the useful indices for continued monitoring, indices such as salinity, TDS, and conductivity were especially useful for indicating riverine inflows. Some of them were more useful at indicating enrichment offshore, such as TDS. But the important thing to note here is that they were most useful when used contemporaneously. Similarly, the zooplankton and phytoplankton biological indices, the carnivore to herbivore ratios, as well as the size fractionation of the chlorophyll A, as well as the total chlorophyll A numbers, they were also useful for indicating enrichment. So in conclusion, based on the suite of parameters, the abiotic and biotic indices, stations such as West Channel and Sansan Bay are recommended for prioritization and continued monitoring in the East Portland Fish Sanctuary. Let's look at my reference, and I'd like to acknowledge the following persons and institutions, and I invite your questions.
Thank you, Dexter Dean, for an excellent talk, and all the presenters in this session. I sort of invite people to continue on questions in the chat function. After consulting with Steve, we've decided that we'll have a short break and reconvene here at 11:25 for the next session that will be moderated by Shelley Arnott on aquatic biodiversity and conservation research. So please, if everyone can come back for 11:25, that would be excellent. Thank you very much, everyone. Excellent session.
Okay, welcome back, everyone. I'm Shelley Arnott, and I'm a professor in the Department of Biology at Queen's, and I'm going to be chairing the Aquatic Biodiversity and Conservation Research session. So our first speaker is Ying Chen, and she's going to be talking about taxonomy as destiny, conflicting phylogenetic trees, and conservation priorities in Canadian chorus frogs.
Thank you, Shelley. I'm gonna share my screen. Share. Okay. Just trying to organize my screen here. Okay. Hello, everyone. My name is Ying Chen. I'm a PhD student in Dr. Stephen Lockheed's lab. Today, I would like to share with you a stunning but also head-scratching story. Taxonomy as destiny, conflicting phylogenetic trees, and conservation priorities in Canadian chorus frogs. Oops. Okay. As many of us are probably aware of, we classify biological entities into the ranking taxa. The science of this classification is called taxonomy, and the basic unit of the taxonomy is species. And from the practical conservation aspect, species is also the unit of management. Indeed, many countries set laws and regulations based on the species, while some countries like Canada and United States also recognize their evolutionarily significant, evolutionarily distinct populations within species. And Canada set designatable unit to manage those populations. So the way we recognize and classify taxa has profound effects on how we manage and conserve biodiversity. In many instances, much of our knowledge about particular taxa comes from morphology. And the problem is that conserved morphology can mask deep phylogenetic divisions. This phenomenon of conserved morphology is particularly pronounced in frogs, where genetic studies reviewed many morphologically cryptic species. Here is just one example, a species group from Colombia in the dark poison frog genus Calastothes. Those frogs, they vary little in size, form, or coloration, yet genetics tells us that they're distinct entities. So the phenomenon of conserved morphology is not confined to the tropical frogs. We also found in frogs in North America. And perhaps the most interesting and challenging are the chorus frogs in the genus Pseudacris, a selection of which are shown here. Brimley's chorus frog, Cajun, Boreal, Western, Southern, and mountain chorus frogs. So all those frogs, they have similar morphology. They all have 3 strips at the back. Their markings, variations are subtle, and their size and shape are quite similar. So we have 4 chorus frogs in Canada, and 2 of which, Boreal chorus frog, Western chorus frogs, are the focus of our genomic study, which I will present today. So our traditional view of the distributions of 2 frogs are based on morphology and call playbacks. So the boreal chorus frogs were thought to mainly found in West North America. So you can find boreal chorus frog from the Southwest US to the Northwest Territory in Canada, over to the James Bay lowlands. And we thought Western chorus frogs mainly occur in the Central North America, and it extends to our Ontario and Quebec. So we are currently here in Kingston, Ontario. And so the traditional view is that we only have Western chorus frogs in Ontario, and we have boreal chorus frogs in West Canada. And a study in 2007 done by Emily Moriarty-Lemon and her colleagues, they used mitochondrial DNA to reveal that we actually have 2 frog species in Ontario. So the red one here showing the distribution of boreal chorus frogs. We have them in West Canada and Ontario and Quebec. And the Western chorus frog is shown in the light green. So the contact zone is here in Southern Ontario. So note that their study is based on the mitochondrial DNA. So frogs are like human beings. We have both mitochondrial DNA, which is maternally inherited, and we also have nuclear DNA, which is biparentally inherited. So this study revealed part of the genetic picture of the frogs. Also note that they only have samples from only a few locations from Ontario and Canada. And so in 2008, COSEWIC, which is Committee on the Status of Endangered Wildlife in Canada, assessed the status of chorus frogs in Ontario. And they found many populations are declining rapidly. And they, in 2008, they considered the mitochondrial DNA data alone to be insufficient to diagnose 2 distinct species. And so they kept the traditional view of Western chorus frog taxonomy in Ontario. However, they did incorporate the mitochondrial DNA into the management decisions. So they diagnosed 2 designatable units. One is the Carolinia, shown in red here, which is not at risk. And the other one is the Great Lakes St. Lawrence designatable unit, which is listed as threatened. And so the fundamental issue here is that we lack data in terms of sufficient intensity of geographic sampling and in terms of having both mitochondrial and nuclear data to address the possibility of hybridization. Basing decisions on mitochondrial DNA alone is problematic because it is maternally inherited and all genes are inherited as a single unit. So the goal of our study was to include genome-wide nuclear markers to clarify the picture of evolutionary relationships across Canada. So the questions we are trying to address is, do we find evidence that both Western and chorus frogs exist in Ontario using both mitochondrial and nuclear data? And if so, where is the contact zone? And do we find evidence of hybridization and introgression? So our sampling to date include over 100 frogs across Canada. We have many samples from Ontario, Quebec, Northwest Territory, Alberta, and thanks to all the collaborators contributing to this project. And so we first review the variation in the mitochondrial gene, cytochrome B, and we constructed median joining networks. And what we found is similar as Emily Moriarty found. So we have boreal chorus frog mitochondrial haplotype, and Western chorus frog mitochondrial haplotype. And notice that the divergence between the boreal and Western chorus frogs in mitochondrial is very deep. So there's at least 87 mutation steps. So there is a deep divergence between 2 frog mitochondrial DNA. And so within boreal chorus frog, we see that there is Western frogs from Western Canada in Alberta and Northwest Territory, and there's boreal chorus frog in Ontario and Quebec. So if we map the mitochondrial DNA into the map, we will see that here the red dots are the Western chorus frog, and the yellow and blue are the boreal chorus frog. So we found the mitochondrial DNA contact zone a bit northward compared to what Emily Moriarty found. So does the nuclear genetic data show the matching pattern? We obtained a genome-wide nuclear dataset using a method called double digest restriction site associated DNA sequencing, so ddRAD-seq. And so we anticipating finding 2 clusters, one corresponding to the Western chorus frog, one corresponding to the boreal chorus frog. And indeed, we found 2 major clusters. However, it is corresponding to the geographics, not corresponding to the species. So here are 2 genetic analysis we did, and the messages are the same. So the first one here, each dot represents each individual. So that if the individuals are clustered together, that means they're genetically similar. If they separate, that means they're genetically different. So in this axis, we have over 96% variation. So the main variation is between the frogs we found in West Canada here and the frogs from East Canada here. There's of course some variation within frogs in East Canada, but the main variation is geographically. So here is another genetic analysis based on Bayesian assignment. So each bar here is one individual, and each color, the color represents the membership, the probability membership to each cluster. So if there are 2 genetic clusters, it will be frogs from West Canada and frogs from East Canada. And if there are 3 genetic clusters, it will be frogs from West Canada again, and some small variations within the frogs in East Canada. So combining both mitochondrial DNA and nuclear DNA, we are actually seeing a discordant genetic pattern. So I found it's very hard to understand at the very beginning too. So I would like you to take a minute to imagine a simple taxonomic scenario. Imagine we have 2 frogs. They have similar morphology, they have similar mating cause, and their mitochondrial DNA are divergent, and their nuclear DNA are divergent. So we would assign or classify those 2 frogs as separate taxa, separate species, because of their genetic differentiations. However, in the real world, in our case, we have morphologically similar frogs. Here, they have similar mating calls. They even have similar nuclear genomes. However, they have deeply divergent mitochondrial DNA. So what are we going to do with those frogs? Are they the same taxa, but they have different mitochondrial lineages? Are they different taxa? They have the similar nuclear genomes. So it is indeed a head-scratching issue. And so in terms of the conservation, how are we going to manage those groups? Do we manage them as a single group or different groups? So for now, our suggestion is to stick with current 2 designatable units. So here, 1 designatable unit, here, another designatable unit. However, we suggest moving the contact zone from south to north a little bit here. So to better understand this mitochondrial nuclear discordance pattern, we hope to create annotated genomes for both Western and boreal chorus frog. We hope to increase sampling in Ontario to better characterize the location and breadth of the mitochondrial DNA contact zone. We hope to sample the entire distributions of 2 frogs, including the frogs in the United States. And in the end, we hope to sample the replicate landscapes to test for metapopulation dynamics to have a better understanding of the approaches we should take to protect the species. So at the end, I wish to thank our founders, including Environment and Climate Change Canada, NSERC, and Bailey Family Chair in Conservation Biology. Thank you very much.
Thank you for a very interesting presentation, Yang. That was excellent. So we have a couple minutes for questions, and you can type them in the chat if you have any. I'll maybe start off with one. What mechanism would cause the difference between mitochondrial and other DNA, nuclear DNA?
Yeah, so that's a very good question. So there's quite a few mechanisms. So a few examples include, so basically mitochondrial DNA is maternally inherited. So you only inherit them from your mom. So one hypothesis is that if the dispersal of male and female are different, for example, males have a larger geographic range and they disperse more, and they were made, while females they tend to stay in the same region. And so when the males mate with females in another region, then the mitochondrial DNA will not be inherited. So that's one scenario. The other scenario is there might be hybridization and introgression, which is simply two groups of organisms they mate together. And so for some reason, the mitochondrial DNA of one group of organism is superior than the other. And so the mitochondrial DNA of that group of organism will introgress into the other. And what we will see is two groups of organism with different nuclear genome, but the similar mitochondrial genome. And so there's also other different hypotheses. But yeah, for the coursework, we are not sure yet. We will have to do further investigations.
Okay, thank you very much. And if anyone has any further questions, just go ahead and type them in the chat and you can answer them later. Okay, our next presentation is by Gina Marie Maddox from the University of West Indies, and she'll be talking about morphological and potential genetic variability in Acropora palmata populations across Jamaica's north coast.
Good morning, ladies and gentlemen. Can you all hear me?
Yes, we can.
Okay, excellent. So my name is Gina Marie Maddox. I'm an MPhil student in the Department of Life Sciences here in the Department of Life Sciences at the University of the West Indies Mona campus. And I will be presenting on some of the preliminary findings of a site reconnaissance undertaken in order for me to properly execute my study entitled The Morphological and Potential Genetic Variability in Acropora palmata Populations Across Jamaica's North Coast. Now, my main points of the presentation will fall under the following headings. I'll be looking at a brief background of Acropora palmata with regards to their ecological significance, the threats, my objectives and tasks, my research questions, the sites that were selected, the preliminary findings, a summary of the sites, the data that will be recorded in the future, and what can be extracted. Now, the Acropora palmata, or the elk horn coral, is a shallow water branching coral that is hermaphroditic broadcast spawner in nature. This really means that it releases both male and female gametes into the ocean. The corals grow about 5 to 10 centimeters per year, and they grow very close to the shoreline in depth ranges of less than a meter to about 20 meters, around an average of 3 to 5 meters. Now, generally, if you're looking at my slide, The Acropora palmatas can grow on the reef flat, or they can also grow on the shallow fore reef. But historically, they were seen on the reef crest, so much so that the reef crest was also given the name the Palmata zone. Now, Guru in 1959 described it as one of the dominant structural framework builders on Caribbean coral reefs, especially because of the functions that they provide. And two of the main functions is a hydrodynamic barrier. They basically absorb the wave energies that are coming towards the coast, lessening the impact from the storm surges during periods of the hurricane season or bad weather. And another important function is that they form dense thickets of monospecific stands, which provide shelters to a numerous amount of marine organisms. Now, the International Union for the Conservation of Nature has listed the elkhorn coral as critically endangered, meaning that there has been an 80 to 90% decline or reduction in the species, as well as its extent. And the other factors that would have led to it being listed as critically endangered is that there has been a decline in the geographic range of the species across the regions where they're originally found in, as well as at least a 50% probability that the species will actually go extinct in the next 3 generations or 10 years. Now, acroporids are being threatened by increased magnitudes and the number of storms, algal phase shifts and in their habitat zone, as well as an increase in sedimentation from freshwater sources and disease outbreaks. Now, some of Jamaica's North Coast reefs, such as the reefs found in Discovery Bay, were studied and documented by early scientists, which created significant baseline information, which basically was able to define the region's corals, not just necessarily for Jamaica alone. Through their studies and more recent research, we observed that the corals themselves have been coming under a lot of stress. They have been directly or indirectly threatened by climate change with regards to the increase in sea temperatures, as well as ocean acidification, anthropogenic stressors, the mortality of the diadema, which is a particular sea urchin that actually is a grazer on the reef, almost like cows, that help to remove and clean up the benthic areas on which these palmatas can then establish and grow from, and as well as other associated phase shifts. Now, if we take a look at the bottom of the slide, I have placed some pictures of some of the unhealthy corals that we have found during our site reconnaissance dives. On the left-hand side, you actually see one where the areas where the orange colors are surrounding is actually a dead patch. The next picture beside it, where you see some green patches, that's actually a recently, what you would say recently dead patch, based on the fact that we can still see some of the green algal turf still on it. The next picture to the right is actually an Acropora palmata colony, which has a disease on it. And the picture to the extreme right is a dead Acropora palmata colony. And that's what we don't want to continue here in our waters. Now, my objectives. Now, Jamaica's Acropora palmata population has never been comprehensively assessed across the island. And due to the important functions, which I had stated earlier on, that it provides to not just the reef or the reef structure, but to the coastal communities and its threatened status, the main objective of my research would then to be to conduct a baseline study of our Jamaica's North Coast population. And based on that, for me to effectively do this, I would then need to map and record locations and the spread of colonies and the population, determine the abundance, the dimensions, and record health indices using transects and structure from motion methods, also determine if Jamaica's elkhorn population displays homogeneity or heterogeneity. Now, what are my research questions? Where are Jamaica's North Coast Aquapora palmata colonies located? What are the different habitat zones in which they are found? What is the condition with regards to the health status of the Aquapora palmata at these sites? Do the Aquapora palmata population show phenotypic or structural variability? And are these differences genetic or based on responses to habitat conditions? Now, my site selection. All of our sites were located, as stated, on the north coast of Jamaica. So especially where you see the stars on the slide, those are areas in which, those are the parishes in which we had conducted our study in. Now, our entire north coast is comprised of what you would call a fringing reef. And the fringing reef would then have what we need, which is the reef crest zone or the palmata zone. In comparison to our south coast, if you take a look at the south coast is a lot wider and would not necessarily have that reef crest zone in which we would need to have, in which we would have to look for, in which we would need to look for our Aquapora palmatas. Now, our sites were selected on the basis of previous research, monitoring by government agencies, fishers and water sports operators, anecdotal applications, and basically deductions based on the type of zone present. Because remember, I had said that historically, our palmatas were located on the reef crest. So if we went to an area and we saw a reef crest, or we saw what we could surmise would have been a reef crest in the water, those sites were actually surveyed as well. Now, a total of 22 sites were visited in total, with Portland, similar to the same parish that Dexter, my colleague, had presented about, had the largest number or the greatest number of sites. A total of 30 hours was spent in the water, as field visits were conducted over a 4-month period. And this was done by doing snorkel and dive surveys conducted by myself and 1 to 2 other divers, when data was collected by general observation and the use of phototransits with the Olympus TG5 camera. Now, field visits were facilitated by NGOs or non-governmental organizations and or fish sanctuaries. Now, some of our preliminary findings. A total of 437 colonies were enumerated. And out of those 437 colonies, of the 22 sites that were visited, Rio Bueno actually had the most colonies present, with a total of 52 colonies, followed by Dancing Lady in St. Anne and Oyster Bay, again, in the same parish of Trelawney. Now, the sites where you're actually not seeing any bar graphs, they actually had, even though they did not have any live coral colonies, they actually had dead palmata colonies, with as much as 40 palmata colonies, especially at this site here in Green Island called Sparkling Water. Now, contrary to what we saw historically in the literature, it's interesting when we look across to our pie chart, where we expected to find the majority of our colonies on the reef crest, we were actually finding the majority of them on our reef flat, with it being 54%. And then behind the reef crest now, the reef flat, we had 32% of our corals being located on the reef crest, and then 14% of the corals being located on the shallow ore reef. Now, it is indeed possible that in the past, the majority of our Aquapora palmata colonies were located on the reef crest, but due to the incidences of the increased magnitude and numbers of storms, we surmise that a lot of the fragments were either tossed onto the reef flat and they established there, or they were tossed down onto the shallow fore reef. Another point that we probably surmised is that because of the magnitude of the waves that are constantly hitting against the reef crest, the time that the Aquapora palmata would have needed to recover after being decimated, say by a hurricane or a storm incident, they're not getting that time period for them to recover. So unfortunately, the aquaporas that we're finding on the reef crest are probably is not the same numbers that we probably would have seen in the past. Now, our health status. The health status of the colonies counted were assessed based on the presence or absence of the disease, dead patches, growing tips, signs of predation, and color of the coral. Now, a Rocklebessa right here, now the unhealthy is seen in orange and the healthy corals are seen in blue. Now, a Rocklebessa showed one of the largest disparities between their healthy and unhealthy corals. Winifred and Monkey Island in Portland had the highest ratio of healthy corals, though Boscobel had the highest total of healthy corals. Based on the zones, interestingly enough, even though the reef crest had declined in the numbers that we were seeing, they had the healthiest corals of the three zones, followed by the reef flats and the shallow fore reef. And our last preliminary finding that we'll be looking at is the difference in morphology.
Well, can you just wrap it up? We're out of time right now.
Yes, wrapping it. I'm so sorry. So basically what we're seeing is that we're seeing three different morphs in the water when we did our assessment, and we want to see whether or not these morphs are as a result of environmental conditions or they're predisposed based on their genetics. And with regards to conservation, because I know this is a section for conservation and biodiversity, the information that will be collected based on photogrammetry, structure from motion, and what we can expect to get from our results is that we would then have some information on our Aquapora palmata, the areas covered in the different bays, the health status due to disease and predators, with regards to our ideal conditions, because a lot of the areas are actually influenced by freshwater outflows, with regards to accessory data for the sites, and especially how to conserve and to rehabilitate. So basically, with my last point, the results can influence and determine what could possibly be the best genotypes and phenotypes to be propagated and used when we are repopulating our reefs. And with that, I'd like to thank my supervisor, Professor Mona Weber, my advisor, Professor Aldo Crocker, my other advisors, Professor Stuart Sandin and Mr. Clinton Edwards, the UWI Center for Marine Sciences, and the Discovery Bay Marine Lab, and the Grace Kennedy Foundation, James Moss Solomon Fund. Thank you.
Okay.
So sorry for going over time.
Thank you very much. That was a very interesting talk. If anyone has any questions, you can put them in the chat, and we'll move on to our next speaker, who is Shrisha Mohit from Queen's University, and she's going to be talking about preventing the spread of aquatic invasive species. What is the efficacy of decontamination practices for recreational watercraft?
Hello, everyone. Whoops, my bad. So I'm just hoping you can actually see the right screen.
Yes, yep, we can.
Okay, great. So, well, hello, everyone, and thank you, Shelley. So today I would like to tell you about my research on decontamination methods that are intended to prevent the spread of aquatic invasive species. So a large portion of this work was done at the Queen's University Biological Station, and I'm quite excited to give you an overview of the findings today. So first of all, we can all agree that freshwater lakes are important for the variety of ecosystem services they provide, for example, drinking and irrigation water, electricity production and recreation, as well as for the rich biodiversity that they support. So unfortunately, freshwater lakes currently face various threats, especially of human origin. So these stresses come in the form of climate change, pollution and various types of contamination. We've got habitat changes, as well as the spread of non-native species, which is what I'll be focusing on today. So the establishment of invasive species in aquatic environments have both severe economic and ecological impacts. For instance, they damage infrastructures and recreational equipment, they affect wildlife, native species, and fisheries, and they can change habitats, food webs, water clarity, in addition to impeding navigation or recreation. It's therefore quite imperative to prevent their range expansion. The invasion process itself consists of 4 main stages with corresponding barriers that invasive species need to overcome in order to successfully establish. So the first stage involves the transport of a species to a new environment, thus allowing it to cross a geographical barrier. So aquatic invasive species have historically been introduced into the Great Lakes via the release of ballast water from ships, but now other pathways include the pet and water garden trade, as well as live food trade and various recreational activities. So in fact, there is plenty of evidence that recreational boating activities are an important vector in the secondary spread of aquatic invasive species from a colonized environment to new disconnected lakes. So aquatic invasive plants and invertebrates can become attached or entangled on watercraft and equipment that are used in infested waters, and they are often capable of surviving transport to new sites. So the recommended approach to manage invasions early on is therefore to prevent the transport of specimens, or we could refer to them as potential propagules, to other sites. So how many of those propagules, their condition or potential to remain viable if released, as well as how often those individuals arrive at a site can all determine whether an invasive species will become established or not. So that's why various agencies worldwide have set or recommended decontamination practices for recreational boaters in order to prevent the overland spread of aquatic invasive species. Here we have an example from the Ontario Ministry of Natural Resources and Forestry. So don't worry about catching all the text on your screen right now. To summarize all the text here, the key recommendations are to clean off debris and mud, allowing everything to dry, and washing with hot water or high pressure. The goal of my research project was to determine whether there is enough evidence to support these recommendations. So first, we reviewed the scientific literature on the subject to find out what is known so far. And well, air drying and immersion in hot water were by far the most commonly studied methods. In general, higher mortality rates resulted from longer air drying durations, or from higher water temperature and longer hot water immersion times. So, as you can see from these figures that show the conditions required for complete mortality from these methods, there was considerable variation in the methodology of the studies, treatment conditions, as well as the species assessed. And so we could not find a clear consensus on best practices. We also identified important gaps in knowledge regarding a lack of studies on other methods and the species assessed. Finally, most of those studies were conducted in settings or with equipment that do not necessarily represent the conditions and tools that are in fact available for recreational boaters. So we went ahead and designed our own experiments to determine the efficacy of specific recommendations, namely air drying outside, testing a range of water pressures, and short exposure to hot water to simulate rinsing. We took into consideration feasibility for the user and included species of relevance to the province. So these were zebra mussels, spiny water fleas, and banded mystery snails for the invertebrates, as well as 3 plant species, all of them having different life histories. So 10 specimens of invertebrates or 10 fragments of plants were used per experimental treatment group. And after the trials, the specimens were returned to tanks of water for recovery. Afterwards, I assessed survival within 24 hours for invertebrates or weekly growth over the span of a month for plants. Well, the first experiment I did was to test the efficacy of hot water. So here I used a multifactorial design where specimens were exposed to 5 different temperatures from 25 to 70 degrees Celsius for either 2, 5, or 10 seconds exposure. And what I found was that increasing water temperature decreases invertebrate and plant survival or viability. As our immersion durations were intended to simulate rinsing, high water temperatures are necessary when the exposure is very brief. So overall, a temperature of at least 60 degrees Celsius would be adequate to produce maximum mortality, complete mortality among the smaller invertebrates and plants tested, but snails were more resistant and required a minimum temperature of 65 degrees Celsius. Next, to test the efficacy of air drying, we allowed invertebrate and plant samples to air dry outdoors under a screened tent for durations lasting from 1 hour to 7 days. These replicate realistic environmental conditions and time frames that recreational boaters would encounter between boating trips during the summer. Here we found that increasing the length of time that aquatic invasives are allowed to air dry will reduce their survival or viability. Smaller invertebrates had 100% mortality after almost 3 days, whereas plants could still show some new growth unless they had air dried for almost a full week. Snails, however, were very resistant to air drying, and we could not accurately determine the exact duration required for complete mortality. In fact, only 48% had died after 6 days of air drying. So far, there has been very little research on combining decontamination methods. If certain techniques seem to work well individually, would combining them actually have even better efficacy? So to test this, we exposed specimens to the 5 hot water temperatures for only 5 seconds, and then immediately allowed them to air dry for 3 hours to 5 days outside. And here we found that indeed, combining decontamination methods improves efficacy. So our results show that shorter air drying times are required if the aquatic invasive species are first exposed to hot water. So again, even if we could not predict the exact conditions for 100% mortality among banded mystery snails, we found that at least we could achieve mortality rates up to 90% if they were first exposed to 60 degrees Celsius water, followed by 4 days of air drying. So already an improvement. And finally, our last experiment was done to determine the efficacy of pressure washing. So here we tested a range of pressures from 0 to almost 2000 PSI. These were delivered using commercially available electric pressure washers, and 0 PSI here corresponds to no washing. We had 2 types of experiments. The first consisted of periphyton, which was grown on metal tiles immersed in the lake for 3 weeks. And the second involved plant fragments, which were manually attached to metal tiles using water-soluble hair gel. We washed the tiles placed both vertically and at an angle on this metal frame that you can see here. And it's meant to mimic the shape of a boat hull while keeping the distance from the nozzle to the surface fixed. For the periphyton experiment, we measured the dry mass of material remaining attached after washing. And for the gel experiment, we counted the number of fragments left after washing. Oops, okay. So in both experiments, washing at high pressure removed more material from surfaces. With proper pressure washing techniques, a pressure of 900 to 1200 PSI was sufficient to remove the most amount of material, regardless of orientation, as shown by the graph on the right here. So the blue line corresponds to the angled surface, and the red line corresponds to the vertical surface. However, it was interesting to note that very high pressures close to 2000 PSI did not produce better results than the 900 to 1200 PSI range. Well, what do these findings tell us? Well, first of all, those results broadly support the methods recommended in Ontario and elsewhere, in that air drying, pressure washing, and hot water work. However, some revisions are required to ensure that recreational boaters are encouraged to apply conditions that will unlikely result in any amount of viable propagules being transported. For instance, the recommendation of 2 to 7 days of air drying seems rather loose when we know that the lower range would only work on smaller invertebrates, some but not all plants, and most certainly not on the aquatic snails. Instead, recommending a longer air drying time in the upper range would ensure that if implemented, a greater number of species and individuals would not survive. Similarly, a pressure between 900 to 1,200 psi instead of 250 psi would be the best to ensure the cleanest surfaces possible. And for hot water, although a temperature of 50 degrees Celsius would result in some mortality among certain species, a more stringent 60 degrees Celsius should be encouraged to ensure very high mortality and to be effective against a variety of species. So recommending the more rigorous conditions might seem like overkill, but recreational watercraft and equipment are often fouled by multiple species where they co-occur. Unfortunately, there is no single technique that would guarantee 100% mortality among all of them, while also being easy and safe for recreational boaters to implement. However, our approach to decontamination can help solve this. So while it is good to perform at least some form of decontamination rather than none, a better approach would be to combine more than one means of decontamination or to implement the more rigorous conditions possible in order to eliminate the most individuals and species and individuals. So if we synthesize both our results and those from previous studies, the best approach might be to implement thorough decontamination methods as a series of steps, each eliminating certain groups of invasive species. So for example, by starting with manual removal of mud, debris, plants, and larger organisms such as snails, following up with pressure washing to dislodge any residue, and next rinsing with hot water, and eventually air drying for as long as possible. So to conclude, we were able to address some knowledge gaps identified in the literature while considering techniques that would be simple and safe to implement. And our findings can also help inform future management strategies in the fight against aquatic invasive species. With that, thank you very much for your attention, and I'll be very glad to take any questions.
Okay, thank you, Shrisha. We don't have time for questions, but certainly people can put them in the chat, and I see that there's one right there. And Shrisha, you can maybe just answer it in the chat. So our final presentation is Wenxi Feng from biology at Queen's. And he's going to be talking about new approaches to monitoring Canadian at-risk species and aquatic biodiversity using environmental DNA.
Okay. Can I be heard?
Yes.
Okay. Thank you. Thank you for the introduction. I'm just going to share my screen right here. Share.
Okay, that's good.
Okay. So hi guys, on behalf of my colleagues and my supervisors listed on the slides, I'm going to talk about, I would like to present some of the work that we have done or is doing in our lab, focusing on harnessing the power and promises of environmental DNA approaches to monitoring species of conservation concerns and aquatic ecosystem as a whole. Okay, so for those of you who are not familiar with the concept, environmental DNA or eDNA is what we call, is found in environmental samples, including water, soil, or even air samples that is released by the organism of interest into their environments. The sources of environmental DNA, including feces, shed tissues, and carcasses, etc. For example, on this slide, in a single water sample from the lake or river or aquatic system, there could be DNA signals from a myriad of species, from arthropods to waterfowl to even aquatic mammals. So detecting presence absence of single species or whole assemblages of species in the environment is made possible by this new technique. So the apparent merit of this technique is that environmental DNA does not require the capture or the handling of the species of interest, and also is very sensitive in terms of detection sensitivity. So it's very useful for working on species of low detection probability using traditional methods and species of conservation concern, because we do not want to invade the species, the organisms. So one of the a few examples of application of the technique, including early detection of invasive species, a survey of endangered species, and since we could get information of many species using the same sample, the description of community structures, even tropical interactions, is also made possible. So there are two. General approaches using this technique regarding your scientific question. The first one on the top is a species-specific kind of an approach where we aim to design assays that are really, really specific to a single species where we only interested in that species, so we can make our detection really specific and sensitive to that particular species. So we can quantify their abundances using this technique we'll call quantitative PCR, a real-time PCR. Another way to do environmental DNA studies is to do environmental DNA metabarcoding, where we design universal parameters, where our assays is much more generous that we can amplify and identify multiple taxa, multiple species at the same time using the same sample. So the sensitivity will be lower, but we have a broader scope of what's going on in the sample. So in the next few slides, I'm going to talk about different projects that using both methods, and start with my work doing my PhD. Then I'm going to creep into others' work just entertain. Okay. So the first study I'm going to talk about is part of my PhD work focused on the northern map turtles. The turtle is quite common in our view station in some of the lakes, but it's broadly considered a species of special concern in the country. And as a mid-sized freshwater turtle, they hibernate in the winter. So for our Jamaican friends not familiar with the winter ecology here, so they hibernate under the water, under the ice for 5 to 6 months a year. And interestingly, this turtle hibernates in large aggregation where we have hundreds of turtles hibernating at the same time, and they keep going back to the same sites year after year. So this hibernation sites could be very critical to their population survival, could be a critical habitat. And the problem to us is, how do we find such habitat without actually being able to go under the ice to see them ourselves? So on the bottom right, this picture is actually we tried to photo them under the ice. But using the environmental DNA, can we find habitat under the ice during winter time? So in this lake, Lake Abinikan, where our station is located at, so in the winter of 2018 and 2019, we took a whole bunch of samples across the lake, showing in the red dots, where we are hoping to find signals of map turtles under the ice. So on this lake, we know there's one site that turtles are hibernating during previous monitoring studies, and there's one site we suspect turtles are hibernating, but we never get confirmed by traditional surveying, like a radiotelemetry method. So among all the sampled water samples, the result turned out we have lots of detections around where we know they're hibernating, which is the red dots showing right here. The black dots are non-detections, and the red dots are detected. I'm going to enlarge the picture here. We know turtles are hibernating around the island, so we have lots of detections around the island, and very pleasing to me and to this work that we find signals at where we suspect turtles are hibernating. And we kind of like we suspect, but we didn't confirm. And to make things even better, we utilize an underwater robot to actually drill into the ice and find this turtles hibernating right here. That's been seen for the first time, as we know turtles in this lake. So the next study is also about turtles, because I just love turtles. And this is also this is another turtle called common mask turtle. It's one of the smallest turtle in the world, about the size of a hand. And then it's also a species of special concern in Canada. And compared to other turtles, this turtle has this unique behavior of not basking. So in the summertime, the turtle will always hide under the water. They will not come up for sunbathing, and making it very hard to find during the peak activity during the year, unless we go scuba diving to actually find this tiny little turtle. So given this cryptic lifestyle, the question to us again is, can we use environmental DNA to locate this turtle? Maybe we can refresh or updating our understanding of their true distribution across this area, given that we're actually living at their range limits in the north. So with that question, in a few summer seasons, we took samples of various water bodies again across the region. So here is where the northern range limits of the species we know across approximately right here. So we took samples at where they are known to occur and where they are not known to be occur. So the black dots are where I took water samples. The red dots are observational data from a public database. So we modeled the distribution, species distribution of the species using data sets that only include observational data and the data set that is complemented or augmented with environmental eDNA detection. So I don't really have time to show the full results of this study, but one quick conclusion we can have is that DNA supplemented data set does change our understanding of the distribution, and the degree of this alteration is affected by the scale we're going to model. So we did models at different scales. So this is just a showcase of how we can use environmental DNA to answer this question. So next, I'm gonna go into my colleagues' work, and first one is the frog. So as Ian mentioned, so we have two lineages of chorus frog in the region: the boreal chorus frog and the western chorus frog. And so one thing Ian didn't really allude to is that it's extremely hard to distinguish them in the field, as they have very similar morphology and almost identical mating calls. I mean, the frogs can tell them apart. They know they are different species, but our humble humans cannot. So it really leaves us the question that how can we detect these two species using other methods? And also their breeding season is very short, lasting only for a few weeks, making the adult frog sampling very challenging. So our solution is that we can sample with water samples after the mating season, where there'll be lots of tadpoles releasing eDNA into the environment. And we are actually developing environmental DNA and qPCR primerase assays to detect presence absence of each lineage across the area, as mentioned in Ian's talk. She has the map, and potentially we can detect contact zones where we will see two lineages co-occur, which means we can detect both species eDNA at some contact zone. And the next study is gonna be on the cyanobacteria. Also, Alan has already kind of talked about this in his talk. And not only they did UAV studies, surveys in dog litter, but also took environmental DNA samples. So cyanobacteria, as Alan mentioned, is a common component of freshwater system. It's everywhere. But whenever there's an algal bloom, there's a lot of dire consequences. The lake will be polluted. The drinking water supplies will be rendered useless, and it will for the ecosystem side, it will block sunlight, deplete oxygen in the lakes, and sometimes leading to the total collapse of the system. So the question is, can we use environmental DNA to monitor and characterize algal blooms? So they have many results to actually present, but here we're going to briefly show two graphs on the right. So they conducted quantitative qPCR to test the overall cyanobacteria and the proportion of toxic strain inside cyanobacteria. So they are good cyanobacteria, they are bad cyanobacteria that is toxic. So they found two things. On top right, the concentration of cyanobacteria in the lake bottom decreases as the lake depth increase, as shown right here. So there's a decrease related to the depth of the lake. And on the opposite trend in the top of the water column, they also found a correlation between environmental DNA and copy numbers, or the quantity of environmental DNA, and the quality of the concentrations of toxins. So if there are more toxins in the water, toxin released by cyanobacteria in the water, the more likely the more environmental DNA you will be detecting. So this work set up a solid beginning stage of using environmental DNA for algal bloom monitoring and also early warning system, combined with his UAV work. Okay, and the final project is also ongoing. What we're gonna talk about is the tree of life eDNA metamarketing aquatic biodiversity. So as alluded by Steve in the opening yesterday, there's a sharp decline of aquatic biodiversity in the past decades, and degradation of freshwater supplies is a huge and pressing issue that we need water to survive. And we are in a unique place in Canada, that we have many lakes and a lot of freshwater reserves. So give you guys a number, in the province of Ontario alone, there are 500,000 lakes on different water systems. So given that we have so many water systems, so we also have lots of issues of regarding aquatic systems, including invasive species, industrial pollution, and also algal blooms, etc. So monitoring such a vast system in real time requires that we we're going to explore new approaches. That is the environmental eDNA. So this is the focus of a new project founded by the Social Sciences and Humanities Research Council, New Frontiers in the Research as the founders. And the idea is how do we monitor such a large system in real time? So first we need to establish the collaboration with a lot of partners to make this happen. So including indigenous and local communities, aquatic ecologists, conservation genomicists, and even aeronautic engineers. So we need a whole bunch of people to actually make this happen. So our aim is to assess richness across multiple taxa and analyze the richness with regard to local environmental variables to create a proxy of environmental ecosystem health, like a living index proposed by WWF in the past years. So with a suite of universal primers for environmental DNA metabolic coding and next-gen sequencing and bioinformatic pipelines, we are hoping to detect a whole range of taxa in real time from a given water sample. And showing here the UAV or the unmanned aerial system can also help us with massive simultaneous sampling efforts. So basically, we can fly a swarm of drones into the field and take samples for us. Currently, we have designed fish primers with capacity of identify and differentiate roughly 300 species, and we will iterate this process for other taxa. So in all, we hope to provide us a relative biomass and species richness database along the Great Lakes and St. Lawrence River. And by looking at the community composition, we want to create indices of current and expected ecosystem health in real time. To summarize this talk, a pack of all this information, environmental DNA tools are enhancing our ability to quantify species distributions and community structures compositions on a community level. And various emerging tools like qPCR, digital PCR, metabolic coding, and metagenomics will continue to complement and help us to tackle this problem alongside with traditional surveying methods. And also involvement of community science and local communities can play a very important role in this project, especially when we standardize and automate this whole process. So with that, we like to thank a myriad of people that is not listed on the slide because we're fear that we're gonna miss somebody on the slide, but we have so many collaborators and founders to make all this happen. So I don't know if I have time for questions, but that concludes my talk. Thank you.
Okay, thank you, Venshi, for a fascinating talk. We unfortunately don't have time for questions, but I'd like to take a minute to thank all the speakers this morning for fascinating talks. I felt like I learned a lot this morning. We're going to take a break and then come back for some breakout sessions. Steve, did you want to say anything about that?
Sure. So I think what we should do is extend the lunch break a little bit. So if it's okay with everyone here, why don't we take a lunch break until 1 o'clock rather than 12:50? So that'll be about 35 minutes for lunch. I realize it's brief, but I've been told that 3 o'clock is the witching hour by which we must be done. So why don't we do that? And we'll come back, we'll talk about the breakout sessions, and we'll move to those. Okay? So have a great, albeit brief, lunch, everyone.